So we're gonna continue today's activities with the research talks. And the first one in line is Dr. Professor Christoph from University of Leeds.
Yeah, such gracias. So as every morning, good morning. So no, that's not the first one. Yeah, that's the first one. Okay. I realized I had to change the title a little bit, but sorry, I didn't. Anyway, okay. So I will. I hope I will not, bore you today with this research that we've been doing for almost 10 years now, that we started in Nice in collaboration with CEA, the Mars Beamline, now you know it, and the ALS as well in Berkeley. Okay, so I'm working in a very different field in which you're used to, obviously, but we are one of the few labs in France working, one of the few university labs in France working on nuclear chemistry and the impact of nuclear activities on the environments and humans. As you may know or not, in France, it's the CEA, the Atomic Agency Commission, something like that, that really takes care of this type of programs, and so we are a very small group working on this subject. And in the world in 2018, there were 450 nuclear reactor in operations and 55 under construction. So you may know, and especially this week, that each year at each COP, now that nuclear energy is becoming an interesting question again, I would say. Whether or not it's good, that's not my point. I don't want to make it my point, but governments, societies, working groups, etc., are now wondering if they should turn back to nuclear energy facing the difficulties we have with producing energy, and on top of it, I should say electrical energy, because the demand in electrical energy is going to be just unbearable for the next decades. Maybe it's a feature you're not aware with, but nuclear electricity today is only 10% of total electricity production in the world. So it's very small. The bad news is the major production electricity is coal and oil, still today. And so obviously that's the bad news. Okay, and also from the history from 1945 to 1980, there were almost 550 nuclear atmospheric tests, especially in the northern atmosphere, and this is a major contribution of artificial radioactivity on Earth right now. Okay. This has been stopped, hopefully, and so this is the legacy from the Cold War. Anyway, so we're working on the impact on the environments and humans, impact on ecosystem, on biotopes of all kind of radionuclides that may be or may become important in case of a nuclear accident. This is not the same picture as I showed you yesterday, but it's the same reasoning as I showed you. This is a picture of, this is a map of Japan. Okay, I like maps, so again, you may have noticed. So this is a map of Japan, and this is a calculation of the plumb of the radioactive dispersion or drift in the Pacific Ocean of cesium after the Fukushima accident. So this is a calculation. Okay, it's not real. All right. And what is real, I mean, where these measurements is all those dots here. In red, you have the cesium-137, and in yellow, the cesium-134. Okay, so even if you're not familiar with nuclear chemistry and nuclear accidents, you have heard probably that the major output of the Fukushima accident was the cesium contribution to the environment and the atmosphere. Tellurium? Well, no. Well, the entire periodic table is produced.
Right.
That's the point. But the major radionuclides which are dispersed and problem is cesium, iodine, tritium. The tritium we don't really care. So cesium, iodine. And then when it becomes to heavy nuclides like plutonium, it's because it has been very bad. Okay, so for instance, Fukushima accidents, a major contribution, like I would say 99% is cesium. Okay. Chernobyl accident is much worse than that. I mean, Fukushima is nothing compared to Chernobyl. Okay, Chernobyl accident was a lot of plutonium and all that stuff. So it's completely different. Yeah. Okay, so it depends on its half-life. So I don't know it by heart, but it's I think cesium-34. We have to check. It's a couple of tens of a couple of decades. So if you have, I think it's maybe 30 years. So if it's a time period of 30 years, you have to count, and that's a rule of thumb. But you have to count about 7 times this period to completely get rid of it. Okay. After one period, you get rid of 50%. Okay. So, yeah.
Yeah, but my question is like
okay.
alpha, the integration, beta, gamma.
Oh, cesium is a beta. Cesium is a beta emitter, so it emits beta particles, but that's not a problem. It emits gamma particles as well. Yeah. But the problem here is not to be irradiated because it's too low. The problem is to live there and being contaminated because you drink water, you eat fish, salads, and etc. And then you accumulate it in your body. So the problem is not irradiation because that's of course too low. I mean, that was a problem in Chernobyl at the first, first days after the accident. But for Fukushima, it's not a problem. Okay, so so you can clearly see that. Okay, so some people can calculate the plumb of the drift of cesium after the accidents. All right, but of course they cannot calculate the speciation of cesium in the seawater. So although they can calculate the drift, their calculation, I mean, it's a good calculation, but their calculation is very how would I would say very basic in terms of chemistry. And actually, in France, we have also calculations like that to try to predict what would be a propagation in case of an accident. But all those calculations don't take into account the chemistry, as bizarre as it could seem, because they can't. It's too difficult. Okay, so they're only based on diffusion with no chemistry. But it's kind of nonsense because the chemistry plays the first role, but they just can't. Okay, so so there is a gap between what you can predict with the most modern tools and what really happens because the chemistry is not known. All right. So we're trying to fill this gap a little bit. All right. So this is diagram that I like because here are some important words. So you have the ecosystems. Okay, so ecosystems are very complex systems with a lot of chemistry taking place in the ecosystems, and this is good for radionuclide, but it's exactly the same for metallic pollution. You know, classical metallic pollution. So it would be exactly the same. So you have a kind of transfer of radionuclides. So we can speak about anthropogenic contribution if you produce those radionuclides and they are not natural. And so they will interact with your ecosystem and they will be transferred, accumulated, etc., and dispersed. Radioecology is a part of ecology that deals with radionuclides. Okay, and so we have to understand this, which we'll never completely understand. Okay, we have to understand speciation. As you notice, I like this word because that gives you the chemistry, that tells you about the reactivity. Okay, but you have also to build up on models, chemical models, physical models, because this is too complex. So you have to play with this triangle in here. Okay. We've been interested in a marine system because, yeah. The purpose of model is just to understand. They are much simpler. You can do chemistry on the benchtop, you know, but chemistry on the benchtop is not representative of what you have here, but it can tell you about speciation. And then from there, you can try to understand simple ecosystems. I take an example. If you want to look at the speciation of iron in a river, for instance, immediately your river is too complicated because it has or zinc. Let's take zinc. Okay, because the river will have iron oxide particles, it will have bacteria, maybe fish in it, and etc. And so it's just a mess. You cannot predict what would be the speciation of zinc discharge, for instance, in this river, any place in the river, because it's too complicated. So maybe you will create a model with just water, and then you will study at the same pH, and we can vary the pH, you can vary, etc., etc. Maybe you will study the absorption of zinc on the iron oxide particles, the natural one. You basically you have a huge puzzle, and you take bricks, okay, and you simplify the puzzle. If not, it's just impossible to do. Did I answer your question? Yeah. Okay. Okay. So yeah, we've been working on seawater. Why seawater? Because seawater is 71% of the Earth's surface. So whenever you have a contamination of something on Earth, it has a good chance to end up in the ocean. Okay, so this is the same idea actually. So we have an extra problem dealing with radionuclides. That's very fortunately the concentration, even in case of an accident, of radionuclide is very low. Why is that? It's because it's dispersed immediately. Okay. So if it's very high, I mean, that's another problem. You just go away. but if usually it's very low because of dispersion, of dilution, of drifts, and etc. and etc. So we have to try to deal with ultra trace scales. So what we call ultra trace scale is below a ppb. You all know what a ppb is, okay? So it's below a ppb or far below a ppb. Okay, so you cannot do the same chemistry below ppb as the chemistry you do on your benchtop. It doesn't look the same. And actually the thermodynamic is not even the same, because the statistics for two atoms to react together becomes very low, okay? And you are never used to that when you do chemistry in your benchtop, because you are working in a glass tube, and there the concentration is always higher than that. So the chemistry is very different in here. And on top of it, you cannot do spectroscopy with such low scale, okay? There is no spectroscopic technique that can tell you what is the speciation at a ppb level. It doesn't exist, okay? You can tell how many atoms you have, okay? That's analytical chemistry, but you cannot tell what is their speciation. So we have to deal with that problem as well. All right. So here is basically all the concentrations of the major ions in seawater. There is the entire periodic table in seawater. It's a very complex system. But as you all know, the major contribution in seawater is chlorine, okay? 0.55 molar of chlorine, and then a whole bunch of your bromine, iodine, etc., etc. Okay. But we are dealing with concentration on that side. All right. So what we do is we dope artificially our system. So we take seawater, we take what we call the natural ecosystems, and we add some contamination. Okay, because if we don't do that, then we don't see anything. We cannot study anything. So this is our methodology. So we work with living species. So we have aquaria, okay, contaminated seawater. It's real seawater. We have living species in it, all right. And so we do some contamination experiments for a time, all right. The duration of the experiment depends, a few days or whatever, okay. And we measure, so we measure a few things. First of all, the species we get here is what we call a sentinel species. This has nothing to do with radioecology. You do the same for environmental studies as well. A sentinel species is a marine species that will accumulate heavy metals very easily. So there are some very well-known sentinel species. Mussels, for instance, are very well-known worldwide. Sentinel species. All species that filter a lot, okay, obviously. Some fishes as well, okay, etc. But the interesting point with mussels is that they don't travel. Okay, they stick at the same place. So they accumulate, so they are an indicator of the contamination at a specific spot, okay, because they won't travel. A fish travels, okay, and so you lose track. All right, there is a factor that I will use in, it's what we call the concentration factor. It's very easy to understand. It's the concentration in the organism divided by the concentration in seawater. Okay, so you have seen some examples during the past 2 days. So so now I don't have to detail that, and so you know exactly how it should look. The first thing we've been doing is try to understand the speciation of pure uranium in seawater, because it had never been done actually, and so that's what we did. And not very surprisingly, so that so the uranium atom is here, okay, and that's carbonates. So you have carbonates ions in here, and the uranium in environment is always uranium dioxo species like that, UO2 2+, it's uranium 6+, okay. So it has a very strong covalent bond in here, and then it has carbonates in there. So that's the speciation of uranium in seawater. Okay. It had been predicted before that, but it had never been observed. So that was a kind of a funny, and that was a funny experiment at the synchrotron, because we dealt with natural seawater, and so when you measure that, if you remember the speech from yesterday, you have fluorescence line from the entire periodic table. And you have a lot of elements in seawater. So you are really blown up by the fluorescence line of all of the elements. So it's a difficult acquisition. Okay, we were working at 5×10⁻⁵, and so you can that's an exact spectrum at uranium at 5×10⁻⁵ in seawater. So it's contaminated. Okay, we did it on purpose. Okay. By the way, if the natural amount of uranium in seawater is around 10⁻⁸ molar. That's the average worldwide amount of uranium in seawater. So we did the experiment, and then we went down to 10⁻⁵ molar, and we have a very crappy exact spectrum. Okay, so 1×10⁻⁵ is not that low for a high sensitivity beamline, but the problem we are having here is the other species of seawater. Okay. Okay, we did the same with time resolved laser fluorescence spectroscopy, and believe me, we ended up with the same conclusion that uranium in seawater is a carbonate species. It's very soluble, so when you go for a swim, you drink it. Okay, so the first living species we looked at is a sea urchin. Okay, so you eat sea urchin in Mexico. Do you? Yeah, you do eat sea urchins in Mexico. No, you don't eat it. Okay, all right. You can eat it. Well, in France we do eat it. Oh, yeah. In Italy as well, they eat it. Yeah, yeah. Uni? I don't know. Yeah. Okay. Anyway. Yeah, you probably. Yeah. Okay. So you can eat it. I mean, you may eat it. Okay. But now I don't eat it after that. All of this. No, not because it's contaminated, but because I'm just disgusted. You know. Yeah. Exactly. Well, yeah. You know, and it's very dirty when they die. It's awful. Okay. Anyway. So good-looking sea urchins are looking that you know, this way, and there are a few centimeters wide. So why did we took sea urchins as a first example? Well, first of all, because again, sea urchins don't travel. I mean, they are very slow. All right. So they walk on the sand, but they don't go very far, obviously, because they are very inefficient to walk. All right. And the second point is it's a very simple organism. Basically, it has a mouth. Okay. A digestive tube, okay, gonads for reproduction, and inside it's just nothing. It's seawater. It's full of seawater. All right. So it's nothing. All right. It's really a basic, very basic animal. It doesn't have any brain because it doesn't have any nervous system. Yeah. It doesn't have a nervous system. And for the little story. Also, we are not authorized to work with animals who have a nervous system. So that's why we took sea urchin as well. So fish, even as little as you can imagine, so you don't really care, you know, little, maybe I shouldn't say that, but you know, the little fish, you know, but you are not allowed because they have a nervous system. Yeah. Okay, so sea urchins, so all the echinoderms and mollusks, they don't have a nervous system, except pulpos.
Oh, yeah.
Yeah, so it's kind of interesting. All right, okay, so we picked up sea urchins, all right, and this is a picture of our experiment. So we put sand so they can feel happy. We refresh the water because otherwise they like fresh water. I mean, even in the Mediterranean Sea. So it's something that you eat in winter, not in the summer. Okay, and so we contaminate this with uranium. All right. And first of all, we try to have a mass balance of uranium in our system, and that's what we have here. And 96% of the uranium stays in the water, which is perfectly fine, okay, because we have an excess of uranium. And so the uptake of uranium with sea urchin is in a mass balance, but water is very heavy compared to sea urchin. You know, its density is much higher. It's very low, okay, but that's perfectly fine. Okay, we did the experiment for 10 days, and then we looked at 3 specimens, okay, and you have the test here, the test here, the gonads here, and the intestinal tract, and this is a quantity of uranium. All right, so you have the intestinal tract in here, the gonads in here, they're orange, and the test in here. The test is the spine plus the cork. So that's calcium carbonate and magnesium carbonate. But I showed you the picture of the spine yesterday. Okay, if you compare with other environmental studies with iron and zinc, that's about the same. I mean, everything goes in the gonads and the digestive tubes, okay? But that's what a sea urchin is. It's a digestive tube and nothing else, okay? So, so we find all the uranium actually in the intestinal tract, the digestive tube, and the gonads. Okay. So that's the first point. Then this picture you have seen yesterday, but I put it back so you have now the entire story. So for the test first, so we have a very low concentration of uranium. We've made imaging on one spine, and as I said yesterday, so I won't come back to that point. We have a smooth, very dispersed, sorption of uranium on the spine, and we know with xenes, very noisy, I agree, but anyway, that the uranium is probably like uranium carbonate. So you have to realize that uranium in our aquarium is uranium carbonate, but the soluble form. So then it sees the spines, which is calcium and manganese carbonates, but it's crystalline. Actually, a spine of a sea urchin is crystalline. It's very hard. It hurts, I can tell you. So it's because it's completely crystalline. So it's very difficult. There won't be any diffusion of the uranium in the calcium carbonate of the spines because it's too ordered as a material. So it only sorbs, and it sorbs as a carbonate species. Okay, then we look at the gonads, and again, you had this picture yesterday. So the gonads are orange. So that's after contamination, we can record an exact spectrum. And then we see a change of speciation. We see a change of speciation between the gonads, I mean, inside the gonads compared to the seawater. I mean, it's not uranyl carbonate anymore. It's something different. And if we look at the image of one gonad cell, as I said yesterday, it's probable that uranium is dispersed on the cell surface. And so if you look in the literature, what is the major protein on the cell surface of the sea urchin? It's a ferritin-like protein. It's a protein that stores calcium. And sea urchin needs calcium for its energetics. And so for some reason that I won't explain, the chemistry of calcium 2+ is not that different from the chemistry of uranium 6+, because they are hard cations. And so it's not that surprising that there would be an affinity between the toposome protein and the uranium. And also we know from other studies that there is a high affinity between transferrin, that, as you know, is a iron protein, and uranium as well. Okay, so everything makes sense. We just need to show it. Okay, so so the toposome is a calcium complexing protein. It's present in the gonads and in the intestines. And about 30% of the amino acid of toposome are carboxylic based, and uranium loves carboxylates. Okay, complexation. Okay, so we thought toposome was a perfect candidate to complex uranium in the gonads. So what we did is we took some gonads, and we extracted all the proteins from the gonads, and we purified the proteins, and we purified the toposome, extracted the toposome, and then we did some coordination chemistry between purified toposome and uranium. Toposome is a transferrin-like protein. You know, it's it's a protein. Yeah, it's 180K protein. You know, it's basically, it looks like transferrin, but it's less order. It's not order. I'm right. You know. Okay. Yeah. Okay. Okay. So so we did some chemistry with this purified toposome. We compare that to the gonads from the sea urchin that have been contaminated. We did exact all that stuff. We did it again and 10 times, and etc. and etc. And we ended up with this chemical model. Of course, I have no picture, but it's because it's just it's an exact model that the uranyl is linked to the carboxylates, so probably to the aspartates and glutamates of the toposome by a mixing of monodentate and bidentate carboxylates. Okay. And this complex is very stable, and so that's why you have to realize that so the toposome displays the carbonates, because uranium comes in as a carbonate species from the seawater, and carbonate is very strong, so you have to be even stronger than the carbonate. So we didn't measure the Ka of the toposome uranyl complexation, but it has to be higher than the carbonate one, so it's pretty high. Okay, in the title there was cobalt, so I thought I should put some cobalt chemistry a little bit. So it's the same system but with cobalt. Okay, radioactive cobalt. So why are we interested in radioactive cobalt, you might ask? It's because cobalt is produced as no, it's like a byproduct, but in a nuclear reaction when you irradiate stainless steel. Okay, okay, and so in any elements that you use for stainless steels, I mean that you use in stainless steel, when it's irradiated, it will produce cobalt-60. Okay, and so it's actually a problem for nuclear ships because of course, you know, there is a lot of and they're on the ocean. So we studied cobalt, all right, and the first question again was you know, what is the speciation of cobalt in seawater? It seems very basic and stupid, but it's not known, all right. And so we did the experiment, and just to come back to discussion from yesterday that you pointed out, I think we had a lot of problem with radiolysis, because, you know, if you put cobalt in seawater, you have a redox with cobalt. It can be cobalt 2 or cobalt 3. But cobalt in seawater likes to be cobalt 2, okay, because of the potential of seawater. All right. But as soon as you put it in the beam, it becomes cobalt 3. And so we struggled with this very simple problem for months and months. We tried to cool it down to, and it doesn't work. As soon as you put photons on it, it turns to be cobalt 3 very slowly, but from one scan to the other one, it moves away and you cannot acquire data. So the only way we solve the problem is actually not to avoid the oxidation, but to make it minor. So because what's easy with seawater is you don't have to buy it, you can take it. All right. So we took liters of seawater, and we make a big reservoir of seawater with cobalt, and we circulate it in our cell. So we have a small peristaltic pump in here, and we cycle the liquid. So as soon as it sees a beam, it's oxidized, but we don't care because we have a large reserve of cobalt 2 that is stable. And so the cobalt 3 is in our measurements, but it's completely minor, undetectable. Okay, so this is the xanes of a cobalt 2, all right? Yes.
Thank you. When you said that it's minor, that means that it's present. So how fast does it change over 2+? Because it sees the beam when I'm not sure how fast you flow, but I would expect that it sees the beam only for milliseconds.
Yeah, so it's not fast. It's really not fast. When you flow it, so you flow it pretty slowly. If not, you have bubbles and all that stuff. So you flow it pretty slowly, and I mean, we had to tune the conditions.
It flows through like a cell?
Yes, so like that, with a peristatic pump.
So it's not a jet that you...
No. It's a very slow flow, but continuous.
Yeah.
Okay. And we measure in the same time. And no, the oxidation is not fast at all. So this way we could have scans over scans that are reproducible.
Yeah,
sure. Okay. Yeah. Okay. So that's the examples. So that's the examples of cobalt in water. So it's cobalt. Cobalt oxygen. Very basic coordination chemistry. 6 oxygens around the cobalt. From again making a link with the first day class, it's octahedron because the pH is very small. Okay. And in seawater, it looks very, very different. And why is that? Is because you still have oxygen in the first coordination sphere, but then you have chlorine in the second coordination sphere. And that's not very surprising because you have 0.55 molar of chlorine in your seawater. Okay, so cobalt is cobalt 2 with a chlorine in the second shell. All right, so this is our sea urchins. This way, since we are working with cobalt, we can work with cobalt 60, which is really radioactive. So we can use ultra traces of cobalt 60. And actually, we can work in becquerels. So we use 500 becquerels of cobalt 60. Okay. To give you a rough idea, that's about 10 to the minus 15 molar. Okay. So you cannot see it, all right? You cannot see the cobalt, but you can see the gamma emission of the cobalt. And that allows you to work at very low concentration that are really environmental-like. And so here is the different experiments with 3 sea urchins again. And we see the same thing as for the uranium, actually. Almost nothing on the test, everything on the intestinal tract and the gonads. Okay, you have some differences between the sea urchins, but basically it's the same. All right. Okay, that's another curve. It's one shot. It's an acute dose. Okay, I mean, and it's this time we did the reverse. We used a macro concentration of stable cobalt, so it's 10 to the minus 4 molar. So it's just below the limit of the DL50 for the sea urchin, so it won't survive that much, okay, because that's a very, very high concentration of cobalt. And we trace it with 100 becquerels of cobalt-60 in order to see it, okay. And so we did a duplicate of that, and so you see you have, so the sea urchin is acquiring cobalt, and then it goes to a steady state, all right. And so it's just accumulating cobalt, and then you have a steady state between accumulation and ejection. Okay, we did the same. We did the exaph of cobalt in the toposome, because now we had purified the toposome, intestinal tract and gonads. And I won't go into the feature of the exaphs, but I will go directly to the complexation of cobalt with toposome. So we were thinking, well, probably the cobalt is also coordinated to the toposome as for the uranium, you know, because cobalt-2 is also a very hard cation. It likes carboxylates very much, okay. Maybe less than uranium, you know, but it's a bit more versatile, but it likes carboxylates as well. So now we did, we tried to learn about the stoichiometry between the cobalt and the protein. And so we did a microfiltration experiment, and then we quantified the cobalt, et cetera, et cetera. And here you have the response curve of the protein as an increasing function of the ratio between cobalt and toposome. And so you end up at a plateau here. So you may, so the result is that the maximum number of cobalt that you can put in a toposome molecule is around here. You know, we don't have a precise number, but it's going to be between 5 and 7 cobalt per toposome protein. So that's the stoichiometry we ended up with. We ask some theoreticians to do a simulation of our what would be the cobalt complex. Okay, so they did ab initio molecular dynamics simulation. So they cannot do the entire protein, and also because we don't know the structure of the protein. So so we asked them just to put aspartates and glutamates in solution and try to understand what would be the coordination sphere of cobalt, and they produce 2 models that are here. And actually, with these 2 models, you have the first. So that's the calculation. You know, the oxygens from the distances produced by the models, and the exaph data is the black line in here. So our exaph data is actually in pretty good agreement with the calculation. So we were pretty happy. Okay, and that's a small movie they made. Okay, so you have the cobalt here. You recognize the square planar symmetry in here. And from times to times, you have an additional water molecule who comes on top of the square planar. So we have a coordination that is going to be between 4 D4H-like and 5 a square planar pyramid. Oh, now I can. Oh, yeah. Okay. We actually, what we did is we worked out a little bit the pre-H with this symmetry and try to, you know, twice check if the symmetry was fine, because for D4H you have a, as you heard in Thursday, a specific pre-H for cobalt, but given the noise of the data, it was difficult to do that. But anyway, we did that a little bit. Okay, so I'm done with the sea urchin. I want to talk you about. I want to tell you a little bit about the mollusk that we did as well. So that's obviously that's a mussel. Okay, a mussel is much more complex than a sea urchin. Well, it's more fun, but it's very difficult because a mussel has it still has no brain. Okay, but it has a heart, and it has gills to filter the seawater. And it has a pancreas and digestive glands that is very small, and it is here. And also a mussel is much more difficult to dissect than a sea urchin. Yeah.
Oh, I heard it has a pancreas. Are there any diabetes type 2 models with this model?
I have no idea. I have no idea. Yeah. But if I remember well, I don't know if I'm mistaken or not. If you look at lobsters, they have a pancreas as well. Lobsters, I think so. And I think the blue color of lobsters come from a copper protein. Hemocyanin, yeah, exactly. Yeah.
Sea snail too.
Yeah. See?
Sea snail too.
Yeah?
Yeah.
Okay. And they,
Pussycorn.
okay, yeah. Yeah, seafood is nice for copper. Yeah.
I've had to extract it before from live. You have to get it from live seafood. So they send you the sea snail and you have to extract it. Want me to repeat that?
Well, it's better if they send you the lobster.
Yeah.
Well, no, let me tell you, in the Solomon lab, at some point they were studying hemocyanin from tarantulas.
Wow.
So they will draw the blood from the tarantula.
Great.
It's like purple and almost sparkly. Very interesting. Anyway, sorry.
Yeah, no, it's interesting. But anyway, I'm discovering the mussel now, you know.
You still eat mussel?
Well, I used to, but no. Again. Yeah. Okay, so that's the aquarium with our mussels, all right? So they are very happy. And so we again, so this is uranium again. So we did a cohort of 83 mussels on the same time. And of course, you have different sizes of mussel. You cannot, I mean, you can try to select, you know, almost the same size, but of course, it's not possible. You know. So what the funny part with the mussel and you know, the sea urchin and all that stuff is that you have to get them. You know, you have to get them fresh. And I'm not a diver, so the first time we were trying to do it ourselves. And it was just impossible. I'm too bad because I don't know how to dive. So, you know, it's like, wow. And then we realized what we had to be a little bit more professional with that. So we went to a marine biology lab, which is not very far from Nice. And they are specialized in marine biology, actually, and marine ecology. And they have divers. Professional ones. So it's very convenient. We order our mussels and our sea urchin, they take their boats, they get it, and then we take the train and then we get the mussels and we come back to Nice, and that's a half an hour train and it's done. Okay. Okay, and they have a small deck and a beach directly on the seashore. So in the summer we can go there, and while we're preparing everything, we can swim. And so here is a cohort of the 83 mussels, okay, and with a size repartition. All right. Okay, so if you look at the concentration factor, so remember the concentration factor is the ratio between the concentration in the mussel and in the seawater. Concentrations factor for uranium are basically pretty small. So you know, it's about the same value, whatever the size of the muscle is. Okay. For the large one, I mean, it's a much higher value, but that's not very significant because we have only 3 points. Okay, but where we have enough statistics, you can tell that you know, the concentration factor is around 10, 15. So concentration factor, just to let you know, is very important because they are the International Nuclear Agency, the IAEA. I was confused between the French and the English one. You know the International Nuclear Agency. Okay, they have edited concentration factors for all seafood, actually, and that's very important and that's very official, because then in case of an accident, and it was very important for the Fukushima accident, you know which species you can still eat or not. Okay, so concentration factor are a big deal, and the international agencies taking care of that. All right. And then we look at the organs. Okay, for the largest ones, so we dissect all of them, and then we looked. So you have the digestive glands in here, the foot, the gill, the body, and the mental of the of the muscle. Okay, so that's muscles from knees. All right, and what you can see is that on the average, all the uranium goes in the digestive gland. All right. Yeah.
Just in the name of moderation, we should wrap up like 10 minutes or something. So
that's perfectly fine. And so the digestive gland is taking up all the uranium. We are working with the naval base in Toulon. Toulon is there are 2 naval bases in military base in France, Toulon and Brest, and Toulon is on the Mediterranean Sea. It's not very far from this, actually. Okay, and it's a naval base that has nuclear you know, the big how do you say it? The big ship where you have planes on it.
Oh,
Yeah. Well, anyway, you see the pictures, you know. Okay. So okay, that's where they are. What's where it is. Okay. And so and so we're working with those people because they use muscles as an indicator as well. Because as I understand, you know, I don't know all that stuff, but they can welcome ships or submarines from friendly countries, of course. And basically nuclear submarines. Okay, so it's a big deal. And the naval base is on city. Okay, so they monitor for any events that could happen. And so we did the same experiment with muscles from this base. Okay, and what you see is that the concentration factors are almost double. Okay, for here, but it's the same here. All right. And that's very interesting because it would show that there is an adaptability for detoxification for those muscles. They are so used, so they are not used to radioactivity, fortunately. Okay, that's not the problem. But in any seaport, you have a lot of contaminants, heavy metals, you know, cobalt, iron, lead, mercury, all that's, you know, dirty stuff. Okay. Mercury maybe not, but lead and you know, and also the base is very polluted from heavy metals because of the ships that were sunk during the Second World War, and so they are, you know, still there, and so, you know, there's a lot of heavy metals there. And so there is probably a kind of adaptation and detoxification. So that's very interesting. This picture, you have seen half of it yesterday. So we we took the gills, the byssus. So the byssus, as I said yesterday, is the filament. Okay, the mantle, hepatopancreas, and here you have the image of the byssus. Here you have the hepatopancreas. So not diffuse, really on the byssus. It's really taken by the byssus, but on the hepatopancreas, it's very diffuse. We did some Zanes. I showed that yesterday. Here again, I won't. Okay. And then we did the exapse on the hepatopancreas just to know again what the speciation of uranium is. It's messy as usual, but we came up with this model again, carboxylate functions, but only monodentate this time, with a protein that we haven't identified. We don't know yet. We did some time resolved laser fluorescence spectroscopy, and that's interesting because we've seen the same. We can compare that with the database and actually end up with the conclusion that uranium is linked to carboxylate species, and probably a mixing of species, which would not be surprising. Mixings of proteins, enzymes, all that stuff, but carboxylates. We also did a fraction separation of the hepatopancreas cells and look at uranium in each fraction. So it's a gross approximation, but it gives you some rough idea. And actually, this is a repartition of uranium, and you see that uranium mainly is in here. Okay. And so the uranium is associated with compartments inside the cells that is also usually associated with detoxification pathways. So that would nicely fit with what we have seen from the muscles from the naval base. Okay, this is my summary. Yeah. And all right, so what I wanted to show you is a little bit of what we are dealing with when we do radioecology, actually. First of all, well, we are dealing with systems that we cannot understand, well, because it's too complicated. And so we have to, as I always say, we have to play tricks and we have to simplify the system. So we are working with ultra trace amounts. And so we have to dope our system in order to understand what's going on. We have to use sentinel species where we can get global concentration factors, but then it's not enough. We want, so radioecologists usually stop there. They get the global concentration factors and that's it. But we want to know the speciation. We want to know where it goes, what are the target proteins or enzyme in order to understand the uptake and understand at some point, maybe what would be a trophic transfer or trophic accumulation transfer for these species. And at the end also as well, we need this data, and that's actually why we get the support from the CEA. We need this data to input some chemistry in their prediction code in case of we call that an event. That's a nuclear event. Yeah. Okay. Yes, I'm done. Thank you.
Any questions?
So for the that was really nice. For the toposome uranium interaction, so just thinking that this toposome looks like you said it's like a transferring type of protein.
It's a transferring like.
So I'm wondering, have you challenged this prep with siderophores or any other molecule that will have lots of oxygens and will like maybe thinking about the bioremediation, like if you wanna. take
Oh,
away
you mean
your-
to compete with the toposome?
Yeah,
No, we haven't done that. We could do much more with the toposome, actually. That's correct. No, but we have worked a lot with transferrin and uranium, and so we have done that with transferrin. Okay, but transferrin, there are two sites that are pretty well defined, although we don't know if the uranium goes on the two ion sides. For toposome, apparently it's completely disordered protein. It has no crystal structure. I mean, not known crystal structure. So we are kind of blind in terms of what would be the interesting coordination sites and etc. But I agree, we could do competition experiments with just to what we are missing in both cases actually for uranium and cobalt is the Ka, the constant, the affinity constant. But that's always, you know, we need to do a dose response curve with the protein, and it's always a long work actually.
yeah, because also you estimated 5 to 7.
Yes, for the cobalt, yes.
It was for the cobalt.
Yeah,
5 to
that's for the
7
cobalt.
For
Here.
the uranium.
Yeah,
you
for the
used
uranium, we didn't do it.
no idea.
Yeah.
Okay.
No.
Thank you.
Another question.
Yes.
Thank you again. Would you infer that foreign metals or contamination are taking part in some biochemical cycles in a kind of biotop? Yes.
I'm sorry, I didn't get the question. So
Would you infer that foreign metals or contamination are taking part in some biogeochemical cycles?
you mean I'm trying to get your point.
Bueno, preguntaba que si esos metales que están ahora, por ejemplo, en un cuerpo de agua o en un suelo que ya contaminaron, entonces, pues van a participar o ya están empezando a participar como en un ciclo bioquímico.
Okay, I got it. Yeah. Yeah, well, okay, so it's actually a good point. Those metals, most radionuclides are anthropogenic. Okay, they've been created by man. All right. So in case of a nuclear accident, what you get is anthropogenic contamination. You know, it comes from men. All right. So most of those, so it's complicated to answer because some radionuclides have no biological cycles because they are not natural. Okay, so uranium is natural, but uranium is not used, obviously, is not used by any biological cycles. It's completely useless in terms of metals in biology. Okay, of course, when you take cesium, radiocesium is anthropogenic, but cesium is natural. So it could replace at some point sodium or potassium in your biological cycles, but being radioactive, of course, it's a problem. Okay, so there are different mechanisms. You have radionuclides that have no role in biological cycles because they are not natural. Okay, but they are radioactive. Okay, so they can replace an element that is very useful for the biological cycles. One example is plutonium, which of course is not natural, has a high affinity for transferrin like iron. so plutonium has no role in the biological cycles, but of course iron does. And so plutonium can compete with iron, okay? And that's of course a problem, okay? For radionuclides that are not natural, but the same element as a natural element, like cesium, iodine, you know, I can take the example of iodine because it's easier to understand. Iodine, radioactive iodine, of course, it's not natural, but iodine you use obviously for the thyroid cycle. Okay, so what's dangerous with radioactive iodine is actually it will replace your natural iodide in your for your thyroid, and that's why you get cancer from the thyroid. Okay, so after the Chernobyl accident, actually one of the major problems was cancers from the thyroid because there was a lot of iodine and they were not prepared. Okay, so some of those nuclides and radionuclides are already involved in biological cycles, and some are not, but they can replace them or compete with them, and that's the question. So it's actually a very difficult question because everything is mixed, you know, and everything is very complex. Yeah, I don't know if did I answer well? Okay.
Do we have more questions?
Thank you for your talk. It was very nice. So, do you know if recently exists any law for regulate or moderate the impact for uranium or for this type of radioactive compounds in France, or which is the most active law in this case to regulate the use for, I don't know, in the case of this experiment?
Yeah. Of course, there are laws about that, and the law is very simple. There is no, I mean, in normal functioning, there is, I shouldn't say no, but almost no dispersion of radionuclides in the environment. Okay, if everything is functioning well, you know, so you don't have an accident, okay, there is no dispersion of radionuclides in the environment. You know, when a nuclear power plant is functioning well, there is absolutely no dispersion of radionuclide. So the law is very strict on that. There's no big, I mean, it's not completely true. I'm just simplifying. There is a very low limited number of becquerels that can be dispersed in the environment by plants. Okay, it's not zero because there is a dilution factor, and you know, okay, so it's not zero, and also the radionuclides will decay. You know, so they can, for instance, disperse tritium in the atmosphere a little bit because tritium has a very low half-life, so it doesn't really matter because it will disappear. Right. But that the problem is not the dispersion of radionuclide in the normal world. The problem is in case of an accident, or as we are all facing right now in case of a military conflict where some countries, you know, might want to, you know, use some nuclear, weapons. Yeah. But that's why the international agency, the Nuclear International Agency, is very, it's really the policeman of the nuclear world. You know, they have they're working a lot on controlling, you know, you know the countries which are, you know, might be problematic, and so they are watching, and that's actually a big huge, I mean, that's more than a big deal, because the entire story between the not doing geopolitics here, but anyway, just a little bit. The entire story between the US and Iran, for instance, is whether or not Iran is able to purify uranium, to enrich uranium to produce plutonium. Okay. So only a few countries in the world, actually, only the countries that sit at the Security Council of the UN are allowed to produce plutonium. And that's only the countries who at that time won the Second World War. Okay. And after that, they decided that nobody else could produce plutonium. Of course, they decided for themselves and for everybody else. So it's kind of, and so that's only 5 countries in the world, right? And then extra countries decided that they were authorized by themselves as well. So they did it. And there are not so many, but some of them, okay. And on top of that, now some countries as well may or may not develop technology to produce plutonium, and that's something that we don't know. Okay, and that's basically Iran and North Korea. Israel does have it without, and that's clear.
Let's thank our speaker.
Thank you.
Our next speaker is so our next speaker is Professor Liliana Quintanar. When she was in high school, she participated in the International Chemistry Olympiad, representing Mexico. And then from that, I guess she got inspired to study chemistry, motivated to study chemistry in the National University of Mexico, UNAM. And I don't know this, but my guess is that she was graduated with some kind of honors. And I assume, and then she did her PhD at Stanford University under the supervision of Ed Solomon, which is a leading figure in bioinorganic chemistry. And then after that, she did a short postdoc in the National University of UNAM. And finally, she became a professor here at Simvesta. And she did a short stay in Boston, right? Also, as a sabbatical. So please, let's welcome Liliana.
Ahí está. Okay, ahí está. And maybe eventually we'll get to the first slide somehow. Thank you. Okay. All right. So I will try to go. Okay, this doesn't work. Oh no, yes, it works. It's countering. Okay, great. So it's kind of weird to be a speaker in a workshop that I'm organizing. So but I will try to tell you, I try to pick some examples, some stories where we have used some XAS, even though my group is not really an XAS group. I remember when I arrived at Stanford and I was choosing research groups to do my PhD. So I interviewed with Ed Solomon, and then I interviewed with Kit Hudson, and then there was a choice of being one student with each of them, or maybe a joint student. But then I went for Ed because I wanted to learn more of the biochemistry and more bio. I have always been more bio. But then Niels came to the group 4 years ago almost, and he basically turned the lab around and said, now let's do a lot of XAS. So the most recent stories, you'll see that we're doing more XAS thanks to Niels being in the group. And I will tell you some stories. All the work has been done at the Department of Chemistry here at Simbastav. Some of you visited the labs last night. But my lab is actually sort of moving now to the Center for Research in Aging, which is in Simbastav campus in the south of Mexico City. And I like always to put this picture for those of you who are not familiar with Simbastav. We're a federal research center. We do research, we do graduate programs in different areas of science and technology. And we have campuses in different places of the country. But right now we're here in Mexico City. So I don't need this slide for this audience because we are all on board of how essential metal ions are for life. But these same essential metal ions like copper, iron, zinc, their homeostasis, all the tight control, all the proteins that traffic them in our body. So this homeostasis of these metal ions is affected in late age, or at least in some degenerative diseases. So for example, in Alzheimer's disease, we know that the total copper in the brain is decreased as compared to a healthy brain. And we also know that the total copper in plasma, the concentrations are increased. And not only that, the speciation of copper is changed. So we have more what they call labile copper that's not bound to these proteins here. And this change in speciation probably explains the different content of copper in the brain. So we don't know why this happens, but we know that it does. And we also know that in many of these degenerative diseases, there are proteins that are metal binding proteins, and that some of them aggregate and form, amyloid fibrils like these ones here that are associated to disease somehow. So we have the prion protein that is known for, prion diseases and mad cow disease, the beta amyloid peptide that aggregates and forms fibrils in Alzheimer's disease, the alpha synuclein in Parkinson's, and all this is happening in the brain. But in diabetes type 2, we also know that IAPP or amylin, it's a peptide that aggregates and forms these fibrils in the pancreas of type 2 diabetes patients. And we also know in cataract disease, these crystallines that are nicely ordered end up forming these non-amyloid aggregates and actually conform the actual cataract that obstructs the light and causes the disease. So what all these systems have in common is that we have one protein or various proteins that end up changing their physiologically normal conformation to a conformation that allows them to form these aggregates associated to disease. And we also know that these proteins in the test tube at least have metal binding sites for essential metal ions, and we're not even understanding what's the physiological and functional relevance of these metal protein interactions. So what we do in my group, and these are the 3 questions that we try to ask in all these systems, is how do these metal ions interact with these proteins? So what's the coordination chemistry? And for that, we use all the spectroscopic tools we can get our hands on. And then once we understand how the metal ion is binding to the protein, how this metal binding event is impacting protein folding and the aggregation of these proteins. Oh, sorry. And also because we like copper very much, and we know copper is redox active and actually one of the metal ions that are involved in oxygen activation. So we ask what are the redox properties of these metal protein complexes. So those are the questions, and we have worked with many of all of these systems in the past 17 years that I've been here at Simbastav. It's been a long time. And today I'm just going to tell you 3 stories highlighting, going kind of quickly on the background, but highlighting what we learned from XAS. So first, we're gonna talk about proteins in the brain, and mostly the prion protein, which you probably know for mad cow disease. That's its dark side. It can form an isoform, the scrappy form that's infectious. It's protease resistant and is the culprit for disease propagation in these encephalopathies that are transmissible. But I'm going to tell you about the nice side of the prion protein, which is usually anchored, normally anchored on the membrane of neurons. And it has a region that's well structured, and then another region that we call intrinsically disordered. So it's floppy. It doesn't have a defined structure. And it's here where all the metal binding action happens. It's been proposed to fulfill many functions, and you'll see later it interacts with many different proteins. It's important for synaptic function. It's important for neuroprotection, neuroplasticity. So it has its good side. Of course, we're interested in this protein because it's been reported first by the group of Glenn Milhauser at UC Santa Cruz, that it binds copper. In this intrinsically disordered region, there are 6 histidines. So copper loves these histidines, they're imidazole rings. And in these, the first 4 histidines are repeats. So this region is called the octa-repeat region. And Milhauser showed very nicely, even with the crystal structure, that copper can bind to each of these histidines in this fashion. And then back then, and I'm talking about like many years ago, when we started working with this, we noticed nobody was paying attention to these other 2 histidines. So we said, let's see what's going on with these histidines. And we do a lot of peptide synthesis in my lab because these intrinsically disordered regions of proteins can be well modeled with peptides. So we looked at these 2 sites, and I won't go over all the details, but we could use circular dichroism, which is a spectroscopy that allows us to look at these metal transitions, like the DD transitions you saw yesterday in Mario's talk, you know, that Tanabe-Sugano transitions. So any electron transition in the UVBs we can see if the complex is chiral. So of course, metal bound to a peptide or a protein, it will be chiral. So we can probe these transitions. And in this case, you know, in the test, you'll figure out how they change with pH. And we realized that copper binding to the histidine 111 is pH dependent with a pK that's right on physiological pH. And by EPR, so electron paramagnetic resonance, that can tell us about the chemical environment around a non-pair electron. I think Thomas mentioned this technique in his first talk. We can get an idea of what is around the metal ion. So we know the environment changes with this pH change, and we know really well, you know, what's the structure of these complexes: copper bound to histidine and amide groups from the backbone deprotonating. So that was really nice. But then we were interested on this site because it has methionines, and some of them are conserved, and we ask what are the role of these methionines in copper-2 coordination and in the redox activity, because I didn't tell you the prion protein binds copper, and when it's fully loaded of copper, it suffers endocytosis. So it ends up in an endosome at low pH and a reducing environment. So we thought, could it be carrying copper, and then once it's inside in a reducing environment, could it stabilize, reduce copper-1 species? So that was the question. And
The finding over there.
this one?
Right.
This one here. Okay, yeah, that one is kind of a little farther. I forget what number it must be 122. Yeah,
Yeah. Okay.
and this is the I forgot to say, this is the hydrophobic region that's known to drive the transition to scrappy.
Okay.
So, and this is why we just work with the peptide. Yeah, but the copper binds here. So we thought these methionines could be stabilizing, you know, they have sulfur. So they, you know, maybe copper-1 would like to be there. So we first probe what happens if we do peptides where we change these methionines to alanine. And basically the copper-2 species are pretty much the same. I won't go into the details, but similar EPR parameters, the same equilibrium, everything seems to be fine.
By CD, also the copper 2 complexes look very similar. So we didn't worry about what these sulfurs are doing with copper 2. We even do calculations with my colleague Alberto Vela here at Chemistry in Simbesta, and got this really nice figure that can actually reproduce this electronic structure, can reproduce experimental EPR parameters within the limitations of a calculation. So we were very happy with the copper 2 description. But then the question, and then we also did some X-ray absorption spectroscopy. Back then, my student Trinidad Arcos got a Fulbright fellowship to go to Stanford with Ed and Kit Hudson. So she was more of a hybrid. So she went with both of them and managed to collect this data and get some XANEs and XFs. And you can see that doesn't matter whether we have the methionines or not, the spectra look pretty much the same. So the coordination sphere looks very, very similar. So we're happy with this. And this is how these complexes look. But then the question is, can we reduce them? So under anaerobic conditions, Trinidad reduced this with ascorbate, and we can follow the copper 2 signal. And indeed, this low pH complex can get partially reduced. Both of them can get partially reduced, although it's easier to reduce this one than this one. But okay, we ask, how does these copper 2 complexes look like? And this is when we stumble against the wall in the lab, because we have no tool to look at copper 1 in my lab. So we look at absorption, circular dichroism, EPR, that's really nice. But then when the copper gets reduced, we don't see it anymore. And this is where XAS comes in. So back then, Trinidad took all these peptides and she prepared, a peptide where you take away methionine 109 or methionine 112, or you take both out. And because we have a pH equilibrium, we did it at low pH and high pH. So this is the data for low pH. And you can see that whatever movement you do with methionines, they all affect. So whether you have one or two, or you take both, the coordination changes. So you see changes in all the spectra. And in collaboration with Claudio Fernandez, our NMR collaborator in Argentina, they saw shifts in the histidine, and also they saw shifts in the methionine residues, on both methionine residues. So our conclusion is both methionines and the histidine contribute to copper 1 coordination at low pH. And we were very happy. But then what happens when we go to high pH? So here, something else, something different happens. When we change one methionine or the other, we see changes. But then when we change the two methionines, it looks actually very, very similar to the other two. So here our conclusion is the copper 1 can accommodate either methionine, not both. But if you take one, it prefers methionine 109. But if you take one, then the other one will kick in. And so now we have a nice picture of what happens when we take these two guys, these two complexes at of copper 2, and we reduce them.
Just a quick question.
Yes.
I remember seeing this data like years ago, and I was seeing it again. Like the copper 1 absorption is pretty impressive if you have the double mutant, right?
Yeah, the blue one. Yeah, it looks very different. Yeah.
So do we have explanation for that? Why it's without sulfur being coordinated more?
It could be. So my theory is that it could do a linear vis-his maybe, and just the rest of the copper 1 could, I don't know what it does. Yeah. Yeah.
Oh yeah, I guess I was gonna. If I could see the rest of it. If I have.
Although you see
not
here,
there.
right? It's not here.
Not there.
Although the blue one does have a little bit.
It does. It has to be
Yeah.
right, but it's
Yeah.
here, yeah.
Yeah. Yeah, back then I had no clue about XAS, like almost zero clue. So we completely trusted, it was Munsarin at Kit Hudson's group. And
Yeah, like looking at the blue line, I don't think there's any history involved in that.
Right.
It's too symmetrical. There's no, yeah.
Interesting.
So it could be, yeah, I have no idea. If okay, we maybe it's the amides from the carboxylic end, because these are amidated and acetylated.
Yeah.
Yeah. Some linear, yeah, copper, nitrogen, something like that. But for us, the important thing was back then is that, okay, we can reduce these two, something we cannot easily do with the other sites in the prion protein. And the prion protein, or at least this site, can accommodate copper-1 coordination species. If you reduce the 4N, you will end up with either complex, so either with one methionine or the other. If you reduce it at lower pH, both methionines can kick in. And I don't have this data, but the NMR spectroscopy showed us they did a titration. So this is consistent, like at here, only one of them is bound, here, two of them are bound. And then eventually, when you go to pH below 5, you personate the histidine, and you end up with a linear copper-1 bis methionine. Unfortunately, I don't think we got to do the XAS of this one, because we learned that later on. And yeah, you know, the student has to graduate, you have to move on. But why is this important? Just like the physiologically relevant thing, two things. Mice, when they are knocked out of prion, they are known to be more susceptible to oxidative stress. And there were some people out there saying that this site, that the prion protein had superoxide dismutase activity. Exactly, that's my faith, right? So we said, this is why we started doing this. Like, okay, to do superoxide dismutase activity, you need to be able to accommodate and actually shift between copper-2 and copper-1 quickly. And this doesn't look that something that can do that, right? There's a big rearrangement of the coordination sphere around the metal ion. So we went out and said, there's no way. I mean, with this copper coordination chemistry, there's no way that the prion protein could be doing superoxide dismutase activity. Now, the other proposal is that because the prion protein undergoes endocytosis, is maybe the prion protein is a copper sensor and carrier. So then we said, yeah, this makes sense, because in the endosome, you will end up with this species, you know, copper reduced, low pH. So a linear copper-1 with two methionines. And then the chaperones that are known inside the cell to take copper are bis cysteines, which is, you know, higher affinity than bis methionine, and they could take the copper. So then that makes sense for the physiological or possible physiological function of the prion protein. But okay, this was long ago, and I have very little time. So I will just tell you that now this is the summary of what we think of about copper-2 bound to the prion protein. So it can bind in 3 different modes in the octarepeat. I didn't go into the details. It can bind to each of these histidines, and now we know this site can be redox active and accommodate copper-1. We went further with copper-2, and then another graduate student, Carolina Lopez, she did some experiments to look at what happens with the prion protein when it's cleaved, because you should know that half of the prion protein in your brain is cleaved like this. It does not have all these binding sites. It only has the histidine 111 binding site. And it's actually not the same as the full protein because it has the free amino group here. So Carolina did a lot of peptide work to look at copper binding. And we found that this site has a very different coordination chemistry from what I just showed you for the full protein. And it has the ability of forming ternary complexes. And we haven't done XAS here. Niels hadn't arrived yet. So this is what it looks like for the prion protein. It can bind one or many coppers depending on the copper concentration at the synapse. It could be more compact or it could be more extended if it has more copper. And if it's cleaved, it will leave out there these fragments that may be copper bound. And it could also form ternary complexes like this. And so it's very dynamic, very diverse. And the next question, which I won't have much time to tell you about, is another side of the prion protein, a good side, is that it's involved in a neuroprotective mechanism to modulate the activity of NMDA receptors. And these receptors are important for memory processes. So if we put in A beta, well, actually not we, the group of Zamponi some years ago showed that if in Alzheimer's disease, when you produce beta amyloid, this beta amyloid peptide has the same effect as a chelating agent, taking away the copper from the prion, and it causes neurotoxicity because it does not modulate this receptor anymore. If you have any questions on this later, you could talk to Victor because I'm rushing here. But basically, it was this hypothesis that the beta amyloid peptide involved in Alzheimer's disease could be competing for copper with the prion protein and causing neurotoxicity. So the next things we've been doing is, well, we and other groups have studied how copper binds to the beta amyloid peptide. And my student Janaí Posadas, who couldn't be here at the workshop, but again, collaborates closely with Victor, and you can talk about this. She confronted each of the different copper prion complexes with A beta and asked the question if this is true, if A beta can take away copper. And the answer is yes. And I won't go into all the details because it's too much information and it's not working anymore. Okay. Yeah, okay. Well, in any case, I was planning to skip it because I don't have time, but in some cases, what she found, and this is really interesting, we're very excited about this, is that the beta amyloid peptide can form a ternary complex with copper and the prion protein. And of course, this is a different model. It's not that it just takes away the copper from the site. Some sites, it cannot take away the copper. Some sites, it can take it away. And in some sites, it forms ternary species like this. So now we're really excited to take these kind of complexes to the synchrotron to get a better structural information about the, how these two proteins or peptides are around the copper ion. And these ternary complexes will, of course, impact the function of the prion protein. And this is just a summary of what I just told you. But then it's not as simple as just taking away the copper. It can form these ternary complexes, but also I told you the prion protein is involved in many functions and it can actually form complexes with many other proteins. So because it's intrinsically disordered in one part, it can really interact with other proteins, and this is important to form these extra matrix cellular complexes. It's important also for plasticity, neurogenesis, cell migration, and many of the regions of the prion protein that are involved in these protein-protein interactions, guess what? They're in the region where it binds copper, in this intrinsically disordered region. So in this opinion article, what we came out to say is, how much of these protein-protein interactions where the prion protein is involved may be modulated or impacted by the metal ion. So how many of those protein-protein complexes could be facilitated or not by copper? And I think we're hoping this will reach out to the neurochemistry community and they will start looking not only at immunoprecipitating complexes, but really figuring out if there's copper there. If not, Victor, we'll get to that one day. So with this, I will end this part. We are interested also on bifunctional molecules with therapeutic potential for Alzheimer's disease to chelate copper, but not mess up the functional part of copper at the synapse. And Victor can tell you all about this in his poster at the coffee break. And also there's a poster by Gerardo. Where's Gerardo? Where's Gerardo? Okay, he's not here. That text him because I'm announcing his poster. Now we're working with tau protein that's also involved in Alzheimer's disease. So I have like 20 minutes for the next two stories. I think I'll manage. Now we'll move on to the pancreas and the amylin peptide. And this is why I was asking about mussels having pancreas. So I don't have to tell you about the importance of this disease. And we are second place worldwide in incidence of type 2 diabetes after the US. It has to do with degeneration of these beta cells in the pancreas that produce insulin and IAPP or amylin, which is a peptide 37 residues. And these beta cells also contain copper and zinc. So we know zinc binds insulin, but we don't know what's up with the IAPP. So back then, when we started working on this, there was one report that copper was inhibiting the amyloid aggregation of this peptide, but that was it. That was all there was. So we said, let's look at this. And it only has one histidine in the whole peptide. And we know that copper loves to bind to histidines. So we first wanted to work with smaller peptides that will not aggregate. So we made these models. And we realized that copper binding happens. So when copper binds to a histidine, it tries to fill up the rest of the coordination sphere with deprotonated amides, as you saw in the prion protein. So it can do it with the amide bonds that follow the sequence or the amide bonds that go before the histidine in the sequence. So this is why we did these two fragments. And we found by CD and EPR that the complex, the main complex, is formed with the histidine and the amino groups or the amide groups that follow the histidine in the sequence, as you can tell here. And I will go a little quickly here. This is PhD work from Carolina Sanchez as well. She did a lot of work. She started blocking the possible sites where copper could be binding. So we thought if it's this amide or this amide, then she blocked them with methyl groups and asked the question, do the spectroscopy change? And as you can see, it changes. Then she also asked, do the serines, these OH groups, participate in copper binding? So she took away the OH groups and put methyl groups. And you can see that she sees no effect when she takes away serine 19. But when she takes away serine 20, definitely the red spectrum is very different from the original black spectrum. Then we also asked if the amide groups of these glutamines could be coordinating the copper as an axial ligand. And as you can tell here, glutamine 21 doesn't have any effect, but glutamine 22, if we make it shorter or take it away, it changes the spectrum. So we came to the conclusion that this will look like a good picture of how copper binds to these. We also did some electronic structure calculations with Jorge Martín del Campo at the National University of Mexico. And we were very happy to see that even though we didn't tell them all the spectroscopy and what were our thoughts, their lowest energy complex, it actually looks like this: copper bound to the histidine, to amide groups, the serine 20. And the not very far away there, complex, it also could have, so if you rotate, I don't have a figure here, but if you rotate the bond here, this carbonyl could also coordinate copper, and that's not very far in energy. But we still were wondering if these asparagines could, glutamines could be participating as weak axial ligands, although those were higher in energy. So this we published some time back with Jorge, and this is our best model. And what I want you to see here is that the serine 20 is definitely one of the important ligands for copper. And then Niels came to the lab and he says, what complex can we take to the synchrotron? And he took, I had forgotten that you took all these. He took the serine 20 to alanine variant and the glutamine 22, asparagine to glutamine 22. So you can see clearly here that the one that's very different from the original complex is the red. So serine 28, serine 20 to alanine. So that's consistent with our model. So we were very relieved, even though all the other work had been published. So this is consistent, right? That the coordination sphere around copper changes when you take away serine 20. And with the asparagine 22, it changes, but very little, so very mild, perhaps consistent with having this ligand as a weak axial ligand. We also see very small differences in the 3D spectrum. And I don't know if we have examples of these news. Okay. Sorry? So Okay, so we don't have examples on this, sorry, yeah,
Liliana,
but at least we see that this is consistent with what we think it is. Mhm.
just to be clear, I see you have the Zanes and then you have the emission intensity. What is that? Where is that coming from?
This is the RICS.
That's
No.
the RICS. Okay.
Yeah, this is the
just,
RICS.
I was like, are we looking? at-
Sorry, I just assume everybody followed all the and since I'm not an expert, but this is the RICS,
Got it.
right?
Okay.
So
No,
this is
that's great.
energy transfer.
Yeah.
And it is emission.
I just wanted
yeah.
to be clear to myself.
But I
Yeah.
mean, the important part is that there's this enormous
Yeah.
shift. That's the important, like, if you look at both.
Yeah,
Okay.
so there's a huge shift in that copper signal when you take away the Syrian 20. And okay, so I forgot, we didn't have the XFs, so maybe one day we'll have the XF and publish this because we actually had these very nice calculations from Jorge Martín del Campo, that if I remember correctly, they're ab initio, so we have trajectories, so this could be some nice correlation to XFs, but yeah, Thomas.
I have a little comment here to your RICS spectra. Another information that actually in your RICS spectra, since it's a copper two plus, the width of your width of your delta one of the LCH light one. It seems to be proportional to the kinetic spectrum. So there might be an additional information in there. We're actually currently working on a paper where we thought we actually see that.
Okay.
And we came up with explanation. Which is splitting of the here actually.
Okay.
And so it does seem that the width actually seems to be connected to the rate.
So we could look at the calculations and look at the.
But you can use it for see how the width is. If one is wider than it's I have to go back.
The more covalent should be wider, no? Because you have
So
more
when it's more covalent, actually when it's more covalent, it be narrow.
narrow. Okay. Why? Because
You know the part about the mark.
we're okay, we can look into
We
it,
took-
but because we have the calculations, so we could correlate.
You can-
If there is a change, we could correlate to the covalency of
Yeah.
the copper center.
In general,
should see it.
when you have covalency, then you reduce or make it narrower. So could be the
Okay.
case as Thomas
Okay,
is
see,
saying.
for the copper is a little different. than what Mari probably thinks because we
Mhm.
have only one hole and the hole is filled. So you have a D10
Uh-huh.
shell. So
And then
actually it is
that's-
transfer
and then
to P here
With
to
the
P,
two P.
make it. Because they got split too, but they make it wider.
So more covalent, yeah. More covalent, well, let's think about it. Of the coffee break. But actually, I think this we collected with you, I think at Isabel, right? Liz? I wasn't sure, so I. What? Old 62. Okay, well, let's talk about this. We don't have the XFs. I just wanted to show you that we did all this work long back and we came with this model and the main message is here in 20 coordinates the copper ion. And we were very happy to see that serine 20 to alanine is the one that shifts most in this experiment. And why is serine 20 important? Because there is actually a mutation in serine 20 that has an impact in aggregation and toxicity of this peptide. And just briefly, so that I don't leave the story incomplete and talk about the aggregation. Remember, I told you copper delays the aggregation. This is how we study aggregation, following the fluorescence of a molecule that reports on this fibril structure. And you can see, the more copper we add, eventually we don't even form fibrils, and it forms this aggregate, amorphous aggregate. And why is this? The nice thing about EPR, and actually this is something else we could do by XAS, is that we could, doesn't matter if it's in solution, right? So we took the aggregates and did EPR, and copper in the aggregates looks very different from copper bound to the monomer. And if you look at the crystal structures of the fibrils of IAPP, we can see that histidine 18, where copper likes to bind, is inside the beta sheet. So here the fibril is growing towards you. So the histidine is hidden. But in the monomer, now we know that it binds like this to the histidine and recruits serine 20. So copper bound to the monomeric form, it's really competing with the conformational change that you need to make the beta sheet structure and have the fibril where histidine is inside the beta sheet. So this explains why copper binding inhibits or delays or the aggregation, the fibril aggregation, and why if you have fibrils that were grown in the presence of copper, the copper ends up bound in a different way, likely in the outer part of the fibril. So different speciation from copper bound to the monomer. And we get the same if we grow the fibrils without copper and then add copper to the fibrils, we get the same signal. I don't have the data here. So in an free energy landscape of the peptide, this is intrinsically disordered. It could form the fibrils, and this is downhill thermodynamically speaking. But copper is actually stabilizing the monomeric form and making it harder, so elevating the activation energy for the conformational change to form the amyloid fibrils. And this is the impact. And now what I think is that this is why copper and zinc, because zinc is also binding to the histidine 18. We haven't done this work, but other people have demonstrated that zinc binds to histidine 18. So my theory now, my hypothesis is that we have copper and zinc in the beta cells of the pancreas to contend to this peptide that if you leave it alone, it could form the aggregates. Now, how are the homeostasis of copper and zinc impacted in diabetes type 2 patients? We don't know much. We know that there's the speciation and the concentration of copper in plasma increases in type 2 diabetes patients, the same as Alzheimer's disease. But we don't know anything about the content of copper and zinc in the pancreas of type 2 diabetes patients. So we would like to look more into this because maybe this is like a good side of IAPP, that it binds the copper and prevents this aggregation. And this is why we have these metal ions in the beta cell. So that was the second story, and I'm not gonna get through. I'm just gonna refer to all the posters on crystalline. The last system that we have studied is the crystalline. So this has to do with the human lens. So the human lens has to be transparent so that light hits the retina. And to do so, these fiber cells are differentiated during the embryonic stage. They have no nuclei, no mitochondria, no organelles, no production of new proteins, and no degradation of damaged proteins. And so the crystallines that you have right now in your lenses are the ones you have for your lifetime. You really need to take care of them. They're alpha, beta, and gamma crystalline. And why do you have to take care of them? Well, if they suffer damage by UV, like this, the tryptophans there will form kynurenine, and this will cause partially folded intermediates that can, lead to aggregation. So this will happen if you go to the beach and you're sipping a margarita and not wearing sunglasses. It could happen. But of course, you go to the beach, sip your margarita without sunglasses, and come back and you don't have cataract disease. This is because the chaperone can recognize, this alpha crystalline can recognize and save it from the aggregation. The thing is that in late age, you run out of this guy. You cannot produce more. And actually, this guy can just keep it out of trouble, but it cannot correct the folding because we're in a low metabolism environment. So you have to take care of your crystallines. That's the message. But when I learned about these proteins, and this is why I like to organize workshops, I met Jonathan King, who he's now retired. He was at the Department of Biology at MIT. But he was actually here in this auditorium 2009. And he came, it was a workshop on protein folding and aggregation because we didn't know how to do this. So we said, let's organize a workshop and learn. And he came and he told us about crystallines. And back then I asked him, what about metal ions? Could metal ions bind these proteins? And he said, you know what, nobody has looked at this. And then, you know, things take time. 4 years later, I went to his lab and we found that indeed copper and zinc can induce the non-amyloid aggregation of gamma D crystalline. So this is when I decided to do a sabbatical in his group to study this a bit further. And I won't have time to tell you all the story, but actually copper can hit the 3 gamma crystallines and form this non-amyloid aggregation. It can do so by causing partial unfolding of the protein. We can follow the beta sheet structure by the circular dichroism, and we see that copper induces these changes and also makes the protein less stable, less thermally stable. So these are denaturation curves. You can warm up the protein up to 80 centigrades, and it just starts to unfold at 80 centigrades. But all these curves are shifted to lower temperature when you add copper. So copper is really decreasing the thermal stability of this protein. I will have to skip this, but we have collaborated with Miguel Costas at UNAM to do differential scanning calorimetry. And this is not reversible, so we cannot get the thermodynamics of the folding, but we can get a kinetic stability. So we can see that the half-life of these 3 proteins can decrease up to 7 orders of magnitude when you add the copper. And we can also know that the separate domains, it is the N-terminal domain, the one that loses folding when you add copper. And this is consistent with DSE because when we add copper, the behavior is very similar to just having the C-terminal domain. So really the first event of unfolding happens in the N-terminal domain. And the chaperone can rescue this, so it can recognize the partially folded N-terminal domain. I will skip this. There's aggregation involving disulfide bridging. Then also Giovanni, who was a postdoc in the lab some time back, he showed that EDTA can reverse the aggregation. So there's some metal bridging component, but not all of it. You see here, we add EDTA and then the thing keeps growing. So there's another mechanism. And then this is the fun part that I wanted to show you. We do EPR and we can see 2 binding sites in each of these 3 proteins, but we don't see all the copper 2. So I told you when copper 2 gets reduced, then we're lost. We don't see it. So we don't see half of, even 70% of the copper. It's gone. So we said, what the hell happened? We thought maybe it got reduced and we saw a free radical in gamma D. So so by then Niels was already in the lab. So he said, let's take these to SSRL. And then also Thomas helped us with this. And in all cases, you see a distinct signal of copper 1, right? That we know already it's an electric dipole transition, 1s to 4p, and it's typical of bicoordinate copper 1. So we could demonstrate that copper 2 is getting reduced to copper 1. Okay, sorry. Okay, this is another story from the EPR. We know it's a tyrosine-based radical. And so this is really interesting chemistry. There's very few proteins that can do this. Under the presence of oxygen, take copper 2 and reduce it to copper 1 at the expense of forming a free radical on the protein. And the question is, why on earth will crystallines in the lens have this kind of reactivity with metal ions? It does have this site, gamma D, that has these 2 cysteines that look like copper 1 chaperones. Remember I told you that copper chaperones that bind copper like this. And we know now from other work, and this is unpublished, I should go really quickly with Rachel Martin, if we take away all the solvent exposed cysteine, there's no more reduction. What's up?
No, look, yes, this reduction. Can you do cyclic voltammetry?
We haven't done that. Can you do it with? I mean, it's a 20 kilodalton protein.
No, it doesn't
You
matter.
could do it, I guess.
Yeah, on the
Yeah.
copper. You know, you say, you do it with you have an exchange of one electron.
Mhm.
You know, I'm not specialist of cyclic photogrammetry, but I think he will show you a nice way
Yeah,
with the typical potential of, you know, your copper two,
Yeah.
copper one, and that, you know, that should be typical of your binding site.
we have done cyclic voltammetry before with our colleague here at Simba with the beta amyloid peptide, 16 residues, and it was a nightmare. The poor student, you know, took 2 years to get the data. So I don't know how easy it will be with the
Just-
pressing. I mean, it's probably doable, but you probably need to put in redox mediators or some, yeah, I don't know.
I think the one problem would be that you need to optimize the conditions such that you don't have aggregation,
Yeah, that's the other thing.
because it's not reversible anymore, and there's nothing cyclic about nothing reversible.
Yeah, that's true. Maybe, yeah, we will need to think about that.
Have we tried doing this in different pHs?
No. Okay, good. So this is a graduate student who's taking over this project.
You mean
No, we
have
haven't
you
done it.
tried to do it in different pHs?
You will try it. Any more questions, Eusebio? So here is Eusebio. You can go see his poster, because on one hand, with Rachel Martin, she has a mutant where she took away all the solvent exposed cysteines, and then there's less reduction, no, and more copper too. And then Eusebio is trying something similar with gamma D, but you know, simpler, glutathionylating the cysteines, and then he is very excited about the latest result from 2 weeks ago at VESP. Yeah, and yeah, I'm almost done. So the beta B2 crystalline, so you go see the poster of Martin. It also is sensible to copper-induced aggregation, and it has an AdCoon-like motif at the N terminal, and it's actually like a buffering, has a buffering effect in the aggregations. He'll tell you all about it. And now we're really wondering why on earth will these crystallines in the lens have metal binding sites? AdCoon sites are typical for copper transporters. This BC site I showed you is typical of copper chaperone. So are these crystallines in any way involved in copper homeostasis? Or is this a vestige from the evolution and the recruitment of these proteins to the lens as crystalline proteins? So that's a question to be answered by, I don't know who, but people who do evolution and, you know, are thinking about these things. But hopefully for today, I convince you that copper and in general metal ions have a role, and specifically copper has different mechanisms that can get activated, and all these together lead to this non-amyloid aggregation. And we are now looking at Cataract lenses in collaboration, this big consortium of ophthalmologists at the public hospitals and National Institute of Medicine and Genomics. My colleague, dear colleague Fanny, which I hope is listening from YouTube, if it's not, if he's still awake, measuring to metal ion levels, and some colleagues in theoretical chemistry to figure out what's the proteomics and the metallomics of the Cataract lens. And I wanted to show this because we would like to do the kind of things that Christoph showed, cuts of Cataract lenses and figure out what's the distribution of metal ions there. Because we do see, this is data by Fanny, the total content of metal ions does change depending on clinical conditions like type 2 diabetes or hypertension and so on. So yeah, we're really interested on how this metal loss of metal homeostasis in aging could have an impact in things like cataract formation and so on. And with this, I will end. I'm very sorry that I ran over time. 5 minutes, I think, or more. Of course, I don't do any of this work. So this is of my students. And I'm very happy to have a very nice group of very enthusiastic students and postdocs. And I showed you a lot of work from past members. And we have very, very nice group of many collaborators here in Mexico, Simba Varunam, in Argentina and in the US. And thank you for your attention. And I should say, this thing gets smaller and smaller in every presentation because there's less and less money in Mexico for science. So at this point, I will probably delete most of those logos. But thank you for your attention. Mm. Mm.
So any questions?
When is the coffee break? Yeah, you get set up. What? Yeah. Can you use the microphone? Yeah, because we have
Bueno,
more streaming.
it's somewhat related. In the making of pharmaceutical peptides, it's very common to put a histidine tail at the end of the peptide because it helps in the purification process. And my question is, do you think a histidine tail of 6 or 10 histidines could be messing up with the copper cycle?
Yeah, if you don't cut it off after you purify your peptide, yes. What you need to do is put the histidine tail and then some cleavage site, and then your peptide. And then once you purify it, also, cut the site. Otherwise, you are administering patients with metal binding, high affinity copper binding peptide.
Thank you.
Yeah. Do they do this? Yeah. I mean, are there any
I think it's-
drugs in the market with this
Oh,
peptide
I don't know.
stack?
It's a very common technique, and
He's from industry.
to use this use this histidine tail.
Uh-huh.
My bet would be that mostly it's cut off, like you said, before you put it in humans, but I don't know.
Hopefully. Otherwise, maybe we can collect a lot of clinical data on the impact of the patients taking these drugs. I don't know. Yeah. So sorry I rushed, but you can go to the posters later. So there's no more questions. We can move on. ¿Vas a usar esto? Shall we let people go?
O sea que
I'm not moderating. Where is my?
everyone thinking now is coffee. Okay, now it's coffee break.
Shall we do coffee break?
Yeah, I think everyone
Come
is
back.
kind of assuming now there's a coffee break.
Yeah, we ran out very late. Hola.
Muy bien, qué padre. No tengas un cargador. Es de estos.
Voy a esta persona puede ver. Algo con no me deja hacer cosas y creo que hay un proceso de esta deja de hacer.
Que está muy lento.
No, es que luego algo cuando está con internet o no con internet, todo esto. O bloquea.
No, me va a pasar. Porque, bueno, ya adelantarme, yo creo que es que van un poquito con un desfase.
De que es de USB.
No, es mini USB. A ver, déjame.
Yo no sé.
Probablemente trae tu teléfono, trae ese estado de cargador. Y está no, pero ¿verdad?
No, yo tampoco.
No, no tenemos. Estoy impaciente por conocer los resultados,
Sí.
pero no sé si los vas a presentar.
Hoy no los voy a no sé. Son pero sí los tengo, ya Liliana y yo los vimos. Tenemos el resultado de eso, ¿no? Pero no son para, yo no los presentaría en el texto.
Sí, ahorita lo cambiaron.
Let me mute my.
We continue now with Nilis Schut, which is a postdoc at Liliana's lab. I will have to apologize that I don't know all the trajectory, but I'm sure it's also very well.
Thank you. Thank you. Yeah, I first want to welcome everyone again and thank the organizers for letting me speak. I have mentioned that, but I repeated I'm a physicist by trade, and I always like to say that I investigate chemical processes and biological samples via physical means. So I understand roughly a third of what I'm doing. And this has never been more true than with this project that we were very happy to successfully finish and publish in PNAS, which is not very often at Similar Stuff. So we are very proud of that. And as you can already tell, it's a pretty international project that we were able to finish during the pandemic or remotely, essentially. So it was there were a lot of challenges, and we all overcame them by working together, and that's always very satisfying. So the title of the publication also talk was Tryptophan Regulates Drosophila Zinc Stores. That sounds, for starters, very uninspiring, but it's actually a pretty groundbreaking discovery. So when it comes down to it, and this, like, I love this periodic table, especially for chemistry. When it comes down to it, like, biology does not need all this fancy stuff chemists are interested in, right? Even of all of these, most of them are just minuscule in comparison to water inside our bodies. But like usually it's carbon, nitrogen, oxygen, phosphate, sulfur. It's like almost all of it, and then there's also something else floating around that makes like nerves work and whatever. And then there are these trace elements in the 3D part of the transition metals, and I do include zinc in that. And whenever we lack any of these micronutrients, this can have quite devastating effects. And the two most abundant ones are iron and zinc, at least in our bodies. And we know a lot about iron and the iron homeostasis and how iron works in the body. And the reason for that is that chemists like iron because it's interesting. It's redox active, unpaired spins, high spin, low spin. You've heard all of these during the past days, right? However, we know very little about zinc homeostasis and how it works in the body, because zinc is none of that. Chemists look at zinc and think, oh, that's boring. Like, that's a D10 system. Like, I can't look at that. It doesn't do any interesting chemistry. I mean, where's all the radicals? Where's all the party going on? So we know, compared to iron, practically nothing of how zinc is being used in biology, except for maybe zinc in DNA or something as zinc fingers, but rather how zinc is transported, how zinc is stored, how the zinc is utilized and chaperoned and whatever. We know very little about that. And if we lack it, is not really a question. Like, a third of the worldwide population actually lacks zinc in their nutrition, and thus suffers from zinc deficiency. And if you go to some regions like Southeast Asia, almost all of them have a zinc deficiency in some extent. That's exactly the point. Zinc deficiency is characterized by, for example, growth retardation and impaired immune function and several other degenerative diseases. However, the most devastating part is during, it usually affects, that's the question exactly, vegetarians, alcoholics, pregnant lactating women, and infants above 6. Why the last 3? Well, we can understand that if an infant is born, it needs a lot of zinc to grow, right? We need it for our DNA, we need it for a lot of processes. And so the mother needs a lot of zinc in order to provide it to the child. And infants above 6 months, this is essentially the breaking point or the point of change where the lactating mother is not capable anymore of providing all the nutrition to the baby. It's still, you can still dar pecho, right? But it's not sufficient anymore. And you have to subsidize or add additional nutritions. And if your nutrition lacks zinc, for example, then you suffer from zinc deficiency, especially in 6 months old. And the impaired immune function due to zinc deficiency leads to up to 500,000 childhood deaths before a year of 5. That's a rough estimate, especially in developing countries. Mexico also suffers from that, but not as much as, for example, Africa or Asia. And the reason is that you suffer from severe diarrhea, which is one of the leading causes of deaths in children, and also of pneumonia, which your immune system is less capable of defending against if you suffer from zinc deficiency. So what do we do against that? Well, especially as we see in television all around, what we do, we collect something, we take medicine against it. So we take supplements, for example, from this company. However, the intake of supplemental zinc compared to other parts of the supplement in these medicines is not very high. It's which is kind of poorly understood. So why are we so incapable of incorporating zinc from these supplements compared to our nutrition, which leads to the goal that we want to advance our understanding of physiological zinc intake and storage. And the best source of zinc, according to CDC and whatever else you want to use as a source, is meat. That's why vegetarians are suffering from that. Why alcoholics suffer from that, we will come to that at the end. That's the zinc part. Now we come to the Drosophila part. And I like to start with a man named Morgan, especially his publication in Science in 1910. And he discovered, and I can't describe it better than he did, but just read it. So in the pedigree culture of Drosophila, which had been running for nearly a year through a considerable amount of generations, a male appeared with white eyes. The normal flies have brilliant red eyes. And that's why the eye color is a very important part of the publication. That's why I want to emphasize that. So we have this, the fly color is usually in red, and suddenly if you wait long enough, a male appears with white eyes. And why is that? Well, what Morgan was discovering here and publishing was the genetics of it. So the white-eyed male itself could be used to breed with red-eyed females, and then was producing red-eyed offspring, male and female, or white-eyed offspring, but only male. And what he was discovering, publishing, was essentially that the gene that was responsible for the white eyes was sitting on the X chromosome. And it was not dominant. So only males were capable of having the expression of the white eyes due to its dominance in the X chromosome. Only that was pairing in both. So he published this in Science, and 20 years later, he got the Nobel Prize for that, because essentially what he has done, and by this work and the follow-up work on that, was he was describing a method that was allowed him to investigate the position of genes by just observation of physiology in his specimens. And this essentially was the revolution for trying to transform biology into an experimental science. Before that, it was just letting stuff grow and looking at it and collecting it in some museum. So
of course, we have learned a lot since then, especially about the white gene and about eye colors. And in order to understand how these colors are being produced, we have to understand how they start. We have two pathways. One is the tryptophan pathway, and the other one involves guanosine triphosphate, GTP. And I'm not focusing too much on this part. We are focusing more on this part. But however, if we have this pathway, we form brown eye pigment and red eye pigments. And in the end, we have our dark red eyes. And what Morgan was describing with the white eyes, which was later called the white mutant, is that at a certain junction, the white gene was expressing a membrane protein, which was responsible for transporting the true precursors of these eye pigments. And thus it was blocking the pathway and the progression of these precursors. And thus there was no eye pigment formed, and thus the eye was white. And then there was another mutant, and I'm just rattling off the mutants now, and I hope that at the end you will understand why I'm just randomly mentioning mutants and eye colors, because actually it feels kind of random, but in the sense it will be important. And then there was another mutant, where they called vermilion, because the vermilion fruit flies showed this bright red, not a dark vermilion color. And the reason was they later found that the vermilion mutation stopped a crucial synthesis of tryptophan into 3-hydroxyquinoranine, similar to the step Liliana was describing due to UV radiation damage, but this time it's actually part of the physiological biological pathways. And thus this is a precursor of the brown eye pigment. Thus there is no brown eye pigment, rather only the red eye pigments. So the mutant is really red-eyed. Okay. And as a summary, we have the wild type, which has the clear, the dark red eyes of the white of the Drosophila fly. We have the white mutant that lacks all these precursors and thus the pigments. And we have the vermilion, which only lacks one precursor or one crucial synthesis step, and only had red eyes. And why am I talking so much about the eye color of Drosophila when I'm actually talking about zinc and XAS? Well, we will come to that in a second. So where's all of this happening? We are in biology. Biology is messy, as also Christoph has showed us, and as we have seen in other talks as well. What I've been shown here, this is turning more and more into biology class, but bear with me. This is the larva before it becomes a Drosophila, right? So it is just this small klein blob you see before it gets all these beautiful fly wings, right? And here again is the tryptophan pathway that is blocked by a vermilion mutant. This is the vermilion gene. So if you have the vermilion mutant, you block this path, this step, this forming of tryptophan into phenylquinoranine. And so tryptophan is transformed into these precursors like quinoranine in the fat bodies of the larva. And then at some point, tryptophan gets introduced into the kidney-like organs, the Malpighian tubules, shown here and here. Sadly, the contrast is not as great as I would like, but it's like this brown color, which is due to the quinoranine inside. And these Malpighian tubules is where the hydroxyquinoranine is then formed, and then this precursor gets transferred into the omochrome, this transferred and then forms the brown eye pigment, right? Here you have already the brown color, it remains brown, and then it goes into the eyes, and the eyes become darker red because of that. And what Fanny's discovered here at Synchrotron, actually a part of it he discovered already in England, and he came here and he keep working on that, is that these organs showed a dramatically high concentration of zinc. Exactly. Like, More like, what? So why are these eye color changes due to a mutation correlating with zinc concentration? Like what? And so could there be a connection between the tryptophan pathway and zinc? And SE is investigating how transition metals are stored, maintained, transported, distributed in biology. This was, of course, piqued his attention. So a lot of talk about eye pigments. Wasn't this presentation about zinc, actually? Now we're coming to the great fun stuff after this introductory course into Drosophila biology. Yeah, you need to be, if you want to do bio XAS, you need to become comfortable in presenting stuff that you have no idea about. And Isabel can, if I tell something too stupid, Isabel can jump at me. This actually. So again, here as a reminder, this is the tryptophan pathway with the part that's taking place in the fat bodies and the part that's taking place in the Malpighian tubules. And what we see here is that's where I essentially came into the project, or that what they were showing me, is that they have these different mutants, the wild type, the white plus the white mutant itself, and then different mutants along the way. The vermilion, they were knocking out K-phase, they were knocking out CN and Kiat and whatever. And they observed in that the zinc concentration in the, I think it's the whole fly in this case, or the Malpighian tube, I'm not sure anymore. That especially the vermilion mutant showed the lowest concentration of zinc of them all. Why is the tryptophan, why is a mutation that affects the tryptophan pathway affecting the zinc concentration in this organs? Was the question. And just to make sure that they are not doing any error, they compared that to iron, and they saw that these mutations had actually no effect on the iron concentration in Drosophila or these organs. And so they thought, well, maybe the secondary question then would, the follow-up question would be, why maybe one of these compounds that are part of the tryptophan pathway themselves are important. So what if we expose the different mutants to the different nutritions? First up is the white plus, which is sort of the perfect control, as it has the most controlled set of genes. And they showed that although the nutrition had a significant increase when the nutrition was when tryptophan was added to the nutrition, other than that, there was no real change observable. It wasn't, at least it wasn't less, right? So more tryptophan in the nutrition, more zinc in the organ, more kynurenine in the nutrition, more zinc in the organ, more hydroxykynurenine in the nutrition, more zinc in the organ, more xanthurenic acid in the nutrition, not more zinc in the organ. That's already very interesting. However, if they use the white mutant as a control, that, remember, it knocks out the capacity of, sorry, that's the more important piece. It knocks out the capacity of transferring kynurenine into the Malpighian tubules. That's what white, the membrane protein of white is doing. And whatever they added to the nutrition then, and absolutely was not affecting the zinc concentration, that was which was already very low to start with. And if they took the vermilion one, so this was just interrupting the tryptophan pathway right at the start of it, they observed that adding tryptophan to the nutrition had no effect, while adding kynurenine, which is after the vermilion mutation, right, to the nutrition actually healed the Drosophila from its zinc deficiency. So this was the first very exciting discovery for them. Tryptophan and its downstream metabolites directly regulates zinc storage in the Malpighian tubules, which still doesn't answer the question why. But it's the first observation. Great. So maybe they were onto something. So they did, I don't want to point here. They did nutritional studies on these Drosophilas, and as they first showed that if they included more and more tryptophan into the diet, the amount of zinc in the Drosophila increased dramatically, right? It goes from, this is the normal amount of zinc you would find in a fly, and they were able to triple this amount from 0 millimolar tryptophan in the diet to 2 millimolar of tryptophan in the diet. However, they were not affecting at all as a control the iron in the Drosophilas. So it's not just a nutritional thing, that's actually a connection between tryptophan and zinc storage. So yeah, zinc concentration is almost independent. However, when they increased, okay, this one is more complicated. So they did the same experiment, but now with zinc instead of tryptophan as a variable. And to further point out that if you have a low tryptophan environment to grow your larva, although you have a lot of zinc in the diet, it still was incapable of surpassing normal levels of zinc after maturation. However, if they had a high tryptophan environment, the flies were easily capable of surpassing normal levels of zinc in their organs, right? While again, as a control, taking iron, it did not affect them at all. So these were the 2 points for them. Zinc concentration in adult flies almost independent from zinc concentration in nutrition. Zinc concentration in adult flies shows linear correlation with tryptophan concentration in nutrition. So what a fascinating observation. Remember, going back to the why does zinc supplementation is not capable of improving, it's capable to helping with zinc deficiency, but not as much as one would expect. Which correlate very nicely with this observation. Adding zinc to the diet is not helping the fly heal its zinc deficiency. Well, however, adding tryptophan to the diet, which is an amino acid that is usually lacking in how would I describe it? In a nutrition, in a diet that is of poor nutritional value, for example, because you are in a war zone or just a very unprivileged society and thus lack a lot of meat in your diet, right? This is already a pretty great observation for that. However, we still don't understand how this connection is done. Why is tryptophan so helpful in incorporating zinc in the Drosophila flies? So dietary zinc concentration during larva growth regulates zinc content in adult flies. And so they, I thought there was another slide after that. I usually have my presentation mode and I know what comes next, but I like it here. So what they did was they now knew that zinc was being stored in, they supposed that zinc was being stored in the Malpighian tubules, and they were trying to understand where, how, and why, for God's sake. So what they did is they did some microscopy on that, either with a green fluorescent protein that was fused to a zinc transporting membrane protein, and then a DNA marking nuclide to see where they're actually at. And as you can see here on the upper part, the vermilion mutant B1 showed that there's zinc being, the zinc transporting protein is being expressed in the membrane, and they were capable of seeing these membranes, these granules being formed. However, they were not fluorescent, because this was the important observation here. If they added quinoranine to the diet of the vermilions, they immediately saw that in addition to the zinc transporting membrane proteins, a lot more of them were expressed in the vermilion mutants. And in addition to that, in the autofluorescent part, they were seeing these autofluorescent granules. And they associated these with a concentration of zinc. So these were zinc storing granules according to their hypothesis. But they were still trying to understand, is this actually zinc inside of them or not? So what do you do if you want to understand where zinc is in your biological tissue sample? We are at an XAS workshop. and they knew Thomas. I don't know, right? You did this with them, I think, Thomas. Yeah, here. And where? So what they just took the Malpighian tubule, which you can see here in the white plus, which is their control, and took them to SSRL and just looked at it in a XRF, so X-ray fluorescence imaging. And here we have the white mutant, which again lacks these granules they were discovering via microscopy, via visible microscopy. And as you can see, although it's easier if you just look at the scale, here we have 575 counts, here we have 11 counts. So there's significantly more zinc in these Malpighian tubules in the white plus in comparison to the mutant that lacks the quinorinine that interrupts the tryptophan pathway, right? And this is also just our unpublished data that I collect together with Christoph at Nanoscopium. We did the same again, but now in higher resolution. And as you can see here, this is the sum spectra of the tissue cut shown here. And if I look at these hotspots here via the K-alpha fluorescence of zinc, we can see these granules. One is in the process of being expressed and one is already formed. And this is the first time we were capable of observing that the zinc granules were being excreted. So this was pretty nice. This was my first time I was doing X-ray imaging, and we're already having such nice results. So we were either very, very capable or very lucky. I think the latter, but still. And but what I essentially came when I first met this project through Funnies, which is sadly isn't here. I hope he's watching. Is huh? Okay, hi Funnies. My question I had for him was like, how is zinc actually being bound? How is zinc bound in these Malpighian tubules? And he was like saying, huh? Like, what does this question mean? Doesn't all metal bind equally? And then we have a bio-inorganic workshop and course after 1 year. It's like, wow, metals are so cool for biologists. So I was having the hypothesis, maybe we are capable of collecting some interesting data via XAS. Just give me some and let's look at it. And as you can see here, this data was collected at Bessie. This is now just the dissected Malpighian tubule. I put it in a sample holder, I put it in the beam, and then I see these beautiful spectra. I was so blown away by that, and it was like, it's dried.
Oh, okay.
So it's just an enormous amount. Like we went 2 weeks ago, we took some similar sample, and it's like all the inorganic chemists are like, oh, 1 millimolar, oh, it's so much, and then it's like 2,000 counts per second. And then I put these tubules inside, and it's like nature having them, it's like 20,000 counts, and like, yeah, nature showing chemists how it's done, right? Nothing about precipitation and whatever solvation. Nature knows how it's done. So we were able to collect these beautiful spectra, but I mean, it's biology is very messy, so there we will show what we are looking at. However, we also took the vermilion mutant, and this is when things got really interesting, because if you see enormous changes in Zanes and exals, then you are already pretty happy, and now we have 2 fingerprints that we can compare, right? And if you remember adding quinorinine to the diet of the vermilion, yeah, I'm sorry, I skipped one part. We also did a protein purification. So we took well, I didn't, Erica did. She took the Malpighian tubules, she dissolved them and purified the protein part of that. And we did 2 sets of experiments runs on that. And as you can see, it's already pretty similar to the vermilion part of the experiment, which was telling us that that the zinc in the vermilion mutant is probably almost exclusively bound to protein, while the zinc in the white plus mutant, white plus control is apparently not bound to proteins, but rather some inorganic compound. And just as a fantastic control, growing the vermilion mutants on a kynurenine diet completely healed or recovered the changes in XAS and XAFs. So essentially healed them completely from it. So zinc is in the vermilion mutants is almost entirely protein bound, and adding zinc completely recovered the inorganic parts of the XAS in the vermilion mutant itself. It was very impressive to me that we were able to show that just very directly while looking at it. So now we know two things. In its physiologically normal state of the control, zinc has to be bound in large quantities to some inorganic compounds. And if we remember the tryptophan pathway, there are some compounds that are immediately jump to mind, right? If we are lacking 3-hydroxykynurenine and xytorenic acid in the Malpighian tubules in the vermilion mutant, then maybe it's all, and we know now that zinc is bound to some inorganic compounds, and maybe zinc itself is bound to these compounds. And this was only possible because we were I was very surprised that we were capable to collect these spectra. So this is a shout out to just trying weird, stupid stuff from time to time and then be amazed by it. Not everything works out, but this definitely did. I am first going to show the result and then I explain it afterwards again. So what we did was we took the spectra from the white plus and the protein purification, and here's the white plus and the vermilion. And what we did is we synthesized zinc complexes of zinc hydroxykynurenine, zinc 3-hydroxykynurenine, and zinc xytorenic acid. And what we could do is we could take the ZANES part of their spectra and the XAFs part of their spectra and recalculate the control spectra using the spectra of the Malpighian tubules that lacked zinc storing granules. So essentially, mathematically adding the spectra of zinc 3-hydroxykynurenine and zinc xytorenic acid was the same as recovering the zinc deficiency in the vermilion mutant via adding kynurenine to the diet. This is the difference between them. This is the protein purification, right? So this is only zinc bound to protein in the white plus. And the black one is the control, which is essentially a healthy, the spectra of a healthy specimen. And the difference between these two can be almost completely explained by the spectra of these two compounds, which is zinc bound to 3-hydroxykynurenine and zinc bound to xytorenic acid, right? And we could either do it, thank you, I agree. We could either do it by using the protein purification or by using the vermilion mutant, and the numbers were roughly the same. Like these are four different spectral composition analysis by just taking 3 spectra and trying to minimize the squared difference to the control spectra, right? Which is the only one. This is which is the one that's equal to that. That's why I was asking you about the ZANES part of your selenium compounds.
Right, yeah. Look at that, those are identical difference.
Yep.
Cool.
Exactly.
Chef's kiss, right?
Yes, exactly. That was a. So how do these compounds? So this was now the first. There was the biological part. Then there was me with the physical part, and then there was Liliana asking about and the chemical part. So do we actually understand? Exactly. Do we actually, like, I was happy with that. Like, wow, this is amazing. And then was like, but then Liliana was like, but she's right. She's completely right. there was how does zinc actually coordinate through No, this is of course only a funny expression. There was actually a scientific discussion going on, and we don't sound like that. You don't, sorry. So how does zinc actually coordinate hydroxyquinoline, and how does zinc actually coordinate xanthoranic acid? And there's a story behind that that usually isn't told. Now we're being recorded, and I'm told anyway. So these were, so when we did that like 4 times, collecting these spectra, and it didn't make sense. It didn't work out. We were sure that this was correct, but it didn't, because the hydroxyquinoline zinc spectra look more similar to xanthoranic acid zinc spectra. And as we later found out, it's how we synthesize it, and I will explain it at the end. So we have now these 2 beautiful spectra, and then we had this wonderful collaboration with Fabiano Nisano from Italy, who was doing NMR spectroscopy, and he was capable of showing by comparing NMR of the free hydroxyquinoline shown here with and without zinc, that zinc is coordinating in this part of the molecule, while for the xanthoranic acid part, it was from, again, with and without zinc, they were showing, capable of showing that this zinc was coordinating in this part of the molecule. And I love this collaboration because it's just NMR is only capable of observing the organic part of your experiment, while XAS in this sense was only capable of observing the inorganic part of the experiment, and both of them were complementing each other so nicely. And this is why this is how the chemists come into play and are really helpful sometimes. And so now we knew where it was binding, and we also knew in what stoichiometry it was binding. Especially for 3-hydroxyquinoline, it was binding together with chloride, 1 zinc molecule, while xanthoranic acid was binding in 2-to-1 stoichiometry. And this is where the funny story is coming into place. If you prepare this compound without chloride in solution, it's not forming in this manner. And this is the important part for us. Like this intensity change, because we are observing a dipole transition into 4p orbitals, is a interesting, can help you gain insight into the symmetry of the compound. And here we have probably a rather tetrahedral configuration, while here, as this is now, these are now split into 2 sets of white line transitions. This is probably rather a more planar configuration, and this is something I've now been observing a lot with zinc. It's either very similar to tetrahedral configuration, or it's rather planar. And the Zane's part is already a very sensitive measure for that. I usually just look at the Zane's here, already can tell you which configuration it's at. Yeah.
Doesn't form any kind of oxyhedral symmetry in the cell.
Yep.
It always just really surprises me that it's only ever tetrahedral or planar. That's
Yep.
something.
Yep.
Look into.
But this is for biologists. This is
Oh.
Mario. I think especially because Funnies is being online, you would like to have the.
Yeah, but I'm looking at Mario's because he's just to know if I'm saying something stupid or not. I would think that because of the crystal fields, it's a D10. You know, it doesn't gain any energy being octahedral. So.
But I feel like in solution, inorganic solution, it does.
Yes, it does. Actually, it does. Yeah.
But it's similar situation that copper 1 and copper 2. So copper 2 is more square planar. And there is an advantage of going Jan Teller, for example. And then in copper 1 with the D10 is tetrahedral.
Okay.
No. But we also had like this discussion with the theoretical investigation. But I'm not sure what the dimension was. What's the variable? It was like zinc concentration in aqueous solution, and then they had more contribution of 4 coordinated zinc in solution, and they were capable of transitioning it into a 6 coordinated zinc in solution. So there's also some variability there. Yeah. But yeah, I have observed 6 coordinated usually in solution. Yeah.
You're citing the zinc chloroquine.
Yeah.
chloride. Yeah. So I guess with nice ligands like chloride that really love zinc, then they like stay there, but then you can with more water molecules kind of transition into a fifth or even sixth coordinate.
Okay.
But
so
but
solvent
really,
exchange
I would
part of
go
this.
more for what Maria said. D10 copper 1 will also accommodate tetrahedral very happily, just like zinc. Yeah, like zinc finger. Fantastic.
That makes sense, the change of ligand and solvent exclusion, yeah.
And if you have something like EDTA, for example, like that, then
So yeah, or just so if you're studying zinc compounds or zinc in biology or anything, and you are at a like if I take samples from our lab to a synchrotron and they first do the copper part and they are like, yeah, there's all this, it's probably 3 oxygen, 1 nitrogen, and it's like very planar and blah, blah, all this information they already have before we go to the beamline. And then we come to the zinc part of them that what do we know there? Crickets, like we know nothing because zinc is hard to investigate by other techniques, but luckily because it's so boring and it's D10, the 4p part of the 1s to 4p transitions already tell us very, very much about what the configuration is without doing any more studies, right? So now I already can tell you this is a more tetrahedral part, this is a more square planar part. And we were able, I did some DFT calculations to confirm that. I already knew the results, so I wasn't surprised, but it's nice to have confirmation, independent confirmation. And indeed, doing the optimization, we observed that zinc bound to the 3-hydroxyquinolinine together with the chloride in a rather tetrahedral configuration. Compare that to the xanthorrhinic acid, which forms a 2-to-1 stoichiometry independently if we add chloride or not to the solution. Whereas was a rather, it's not square planar, it's rather square planar, right? Due to spheric hindrance, it wasn't forming an actual square planar configuration. But it's close enough to confirm my suspicion I had already from looking at the Zanes. And then, of course, I did all of that at once, but it's just better to tell it that way. I did confirm that by comparing the distances from EXAF's analysis of these measurements with these distances. And now the keen eye will already look like it's, I show it to you with the Debye-Waller factor because you are an educated audience now, right? And as you can see, Debye-Waller factor are in a reasonable realm and the distances agree quite nicely. It's not perfect, but DFT and EXAF, there's already always some difference. However, especially the xanthorrhinic acid spectra had to include and rather impressive, there's also a third sphere here that's also necessary to fit in order to nicely reproduce these spectra, which at large K disappear due to that reason. I lost no one. There's a question over there. Wait.
The use of DFT in this example is for getting the distance after the, how do you say that? Optimization of the structure.
Yes, it's about the distances, but it's also about the general confirmation of it. Like I already, if it would have been more planar, it would have been kind of at odds with the shape of the white line shape from Zanes, right? So there were 2 sets of information that need to be confirmed. So one, we knew from NMR where it would bind, but NMR doesn't tell us in what coordination it binds, right? So this we got from DFT, and it agrees nicely with the spectra collected at the synchrotron, the Zanes part as well as the EXAFs part. It's both that are, and that's usually you cannot use Zanes in such a direct way, in my opinion, but for zinc, it's actually pretty straightforward because you can neglect most of most of unpaired 3D electron contributions. It's just 4p.
So I don't understand why it's not completely planar. Is this some sort of Jahn-Teller effect? Or?
So at least in DFT, we had this hydrogen bond between between one xanthoranic acid group and the carboxylate of the xanthoranic acid. And this was kind of distorting it. And it's also just plain not really possible to perform perfect planar configuration because it has this steric hindrance of its own dimension.
But you have two identical ligands, right?
Yes.
So in theory, it could completely.
No, because there's actually a repulsion between itself, between the one chain end and the other molecule. So it would only be capable of forming a perfect square planar if there would be nothing in the way. However, this hydroxide, I don't know what's the group name for that, is actually sterically hindering itself from being completely square planar.
I was just going to say it's possibly evolutionary, not really sure why you will never see perfect symmetry in a biological ligand set. You just won't. There's always something that it does better than us in some ways, I think, for optimizing. And I think it's just the billions of years. But yeah, you'll never see perfection in terms of symmetry.
Nils, why in the xanthoranic acid zinc do we have a zinc to carbon distance? So,
because we have here carbons.
But they are linked direct to the zinc, or they are in the second sphere of coordination?
This is the second sphere, yeah.
Okay, yeah, thanks.
It's just this is the second sphere contribution, and because the compound itself is so stable, these appear as a very obvious second sphere in XRs.
Okay, thanks.
So it was never considered that that can be like an ultrahedral with an elongation distortion, because it has the oxygen zinc distance of nearly 3. So it may be like a very weak coordination bond.
But this wouldn't neither agree with NMR, nor with Zanes. So yeah. And there's so could you have, for example, I don't know, like 2 oxygens, something like that? Could you, like, this actually can form an almost octahedral configuration depending on pH in the computer, but we were incapable of observing that experimentally, no matter what we did to it. So yeah, there was a consideration of having potentially a 6 coordinated one, but we weren't able to observe it experimentally. Yeah. And as we were learning more and more and more, is that zinc is just not really interested in that. And this is why we couldn't reproduce it experimentally and only in the computer. This is at least my point of view. Christoph. There's
just remind me, at Nanoscopium, when we've recorded the zinc image, you don't have the Xanes of that.
We have a Xanes of that, and this is now very current investigation. So we're unsure about. There's a lot of really interesting open questions we are now addressing. That's why we also went to Bessie last week, and we will. Well, I won't, but they hopefully will return to Nanoscopium because it's a perfect opportunity to answer, especially this question.
Yeah, yeah. Okay.
Because we have these 2 fingerprints, right? And we have the 2 compounds, and we can compare the 2 compounds to the biologically collected spectra, right? And this is exactly where I want to go back to the initial image, right? So this is the fluorescent counts of the K alpha, and at Nanoscopium, as they are collecting with a multi-channel analyzer all the fluorescence, and so I can just as well look at the chloride, and there's at least a hint of a coincidence of chloride and zinc, but this needs to be confirmed further and confirmed by going again and having this. This was the first time. It also pretty impressive. But yeah, maybe this is a hint that the because as you see, the EXAFS part is just as you told us in with the problem in dimension, like the spot itself is larger than the organ itself. So we see all the zinc in the entire organ, but the granule itself is like 3 micrometer in diameter. So we are like from EXAFS or NMR or the whole zinc concentration, we are incapable of showing that all the zinc is actually in the granules. It's just coincidence, like well, it's very likely not coincidence, but we are not showing that there's actual that the zinc is actually in the granules bound to these molecules, right? And this is exactly the question we would like to answer in the future at, for example, nanoscopy. So yeah, this is where there's actually a comment I want to go back. So going back to the part of the zinc deficiency risk groups, vegetarians we understand now, because tryptophan is just not part of a vegetarian diet, at least not the hipster part of the vegetarian diet, right? But once you understand maybe what you should look out for, and if you are at risk of zinc deficiency, maybe you can adjust your diet in order to incorporate more tryptophan. There are vegetarian dishes that have like nuts, for example, that have a lot of tryptophan. However, if you, for example, look at corn, maize, so tortillas are actually very poor in tryptophan, which could be an explanation why it occurs, zinc deficiency occurs, especially in impoverished parts of society in Mexico. Huh? Not much. Like it's usually, I think if I remember correctly, it's like nuts have more tryptophan and zinc, especially. Yeah. Alcoholics actually is very interesting, maybe in the future, because they show alcoholism affects the tryptophan pathway as well and kynurenine levels in the blood. So this is kind of cool as a coincidence. And pregnant and lactating women and infants, we already understood previously, but it could help in treating, better treating the effects of zinc deficiency in, for example, during a famine. And one thing that's been done for infants, especially like if you see what the UN is doing in order to help refugee camps, what they are doing is they use a peanut paste which they give the children for a week. So which could be a good explanation, again, peanuts richer in tryptophan and also in zinc. Why this helps a lot in preventing pneumonia and severe diarrhea in infants treating with this nutritional solution. Okay, and that's it. I would like science is collaborative, especially in synchrotron spectroscopy. So there were a lot of people involved, a lot of people you have to thank, especially Fanny's, of course, for having the curiosity at the start, and then for reaching out to us here at Simbistaff, and my former connections with Europe from this is my group where I did my PhD, which kindly supported us, and as well our friends from the Pauchera Institute, which were also helping along a lot during the pandemic, doing remote experiments with us, and Daniele and Fabio and Alessandra, yeah, sorry, I blanked there, in Italy, who were like a tremendous help in for us better understanding the inorganic part of the chemistry. And here are some photos from Soleil, and Fanny is connecting remotely and making sure that we are collecting good data. Yeah, and so exactly, exactly.
News, I got two questions. The first is the spectra that you got from the XAS was like 70% corresponding to the uranine complex, and the other 30 about with the santorenic acid, right?
it's like it's 70% of the like reproducing the control spectra and required 70% of hydroxyquinone bound to zinc and roughly 15% bound to xanthoranic acid and zinc. But this is just the first result, and there's now like this whole like actually having this inorganic connection between a previously completely independent tryptophan pathway and zinc pathway is very exciting, because there are a lot of degenerative diseases as well as nutritional diseases that have previously been independently connected to, for the example, the tryptophan pathway or the zinc pathway. But of course, having now a reason to connect them on a chemical level might necessitate to rewrite or reassess a lot of these information. So that's why it's so groundbreaking in a lot of senses for hopefully years to come.
Okay, and my second question is, have you tried get your flies drunk?
No, but yeah, maybe. Can we get alcoholic Drosophila flies? Yeah,
that will be it.
We can do the experiment this evening. That we go to Garibaldi. Isabel, bring some flies. Very nice talk, Niels. I just want to read some comments from Fanny's on the chat because he's watching and he's probably eager to have the microphone, but Fanny's, you're not here, so I'm going to read your comments. Niels, a physicist, has done a wonderful description of Drosophila genetics. Impressive. So happy to see the interdisciplinary science going both ways, physics to biology, but also biology to physics. I will just say, add to that, going through chemistry. and then he says the amazing sentence Niels wrote, in essence, adding spectra from 3-hydroxyquinone zinc and xanthoranic acid with zinc to a spectra from samples lacking zinc storage granules has the same effect as adding quinurene into the diet of B1 larva. And I don't see any more comments, I think. So I just wanted to comment. Thanks.
And maybe having all of them here together is like think of my initial sentence: the physical method, biological part, chemical part. If you separate each of these each of these contributions, they don't help as much. Like if you have the biological experiments would have not been capable of actually connecting zinc and the metabolites, while the chemical part would not have been able to connect them to the biological sample. Like the synchrotron part was this essential bridge that needed to be built between these two. And this like that's why I love this project so much. So it's this amazing collaboration between fields and expertise that made these discoveries possible.
And for those of you who are not synchrotron users yet and think like, why the hell will I be in a workshop about metals in biology and synchrotron? You probably should know the story that I really had to almost beg Fanny's to come to LAVIC 6 years ago. And that's when he met Thomas Kroll up here. And then Fanny went crazy about synchrotrons. And he's asking on the chat, have we applied for beam time and nanoscopium?
No, not yet.
Thank you very much for the talk. It's a great example of interdisciplinary experiments. And my suggestion is, have you done or tried to done or planning to done these compounds with cobalt-2? Because it may be stabilized the same geometries, and you can have more spectroscopy to study them.
So no, but you are indeed on a good path there. Like the Malpighian tubules themselves have a rather high concentration of a lot of metals. Like now we only showed like zinc, and we also showed iron as a control. But there's now like nature. Either nature has a very specific reason to be very particular about the use of a compound, or it uses as much of it as possible in any possible way. And that's of course, if we are now like these metabolites were previously not being investigated in terms of metal chelators in a way, right? Or essentially metal binding metabolites. And now this is an open question, like how much can it bind? What else could it bind? If it's, why isn't it binding that, or why maybe it is binding that? And yeah, so no, at least not ready for showcasing here.
So let's thank again Niels for the wonderful talk.
Thank you.
And now we have another talk from Kelly.
Also, I have some leftover mantecadas if you're hungry.
No? Yeah. Oh, and this, yeah, is for that, exactly. And then the Zoom, no? Yeah, you're a good moderator for me. You'll be able to tell me, come on, stop. Okay, so tú puedes poner el código.
No lo tienes tú, ¿verdad?
¿Es el mismo? No sé. A ver. Yeah, I think so. Voy a poner una alarma. 2 horas. Yeah, that's good. Oh. We're waiting for the password. One, two, three, four. Oh. Okay. Still waiting on the password. Well, while we're waiting, I can check that this will advance. Hmm? What do you mean? It's okay. Well, he put it here, but I think. Yeah, good. Okay. Great. Oh, cool. Yeah. Okay. I love that. Okay, we're good. Just need the passcode. Okay, and then got it. See that? ¿Dónde? Okay. Okay. It's okay. Let's say starting.
So let's welcome back Kathy. We have already encountered and started to enjoy a lot of her positive energies you bring to the workshop. And see her
Locura.
complete her talk from
Yes.
yesterday.
Yes, and I'm going to time myself for the science so that I have plenty of time to address the other stuff. So 15 minutes. Okay. So thank you for having me once again. To those of you who missed yesterday's talk, I'm sorry, because we're going to have to go quickly through some of the processes from yesterday to describe the continuation of the research that I talked about yesterday. You'll see. So this is going to be about how you complement bio-XAS with other methods. And this really will make sense once I get into the rest of this. So okay, fine. So the take home of this 15 minutes here, we're going to talk about the CUS system. So we're going to talk about the gold and silver transporter briefly. And we're going to talk about, this is the take home. We use a combination of XAS, of course, and rapid fluorescence spectroscopy in lab, as well as some other things, to understand more of how two of the proteins involved in copper efflux interact, the CUS-F and the CUS-B, the little green one that was running around and the little blue part, because I had told you these interesting things about how they interact. And what we found was evidence for the rate of metal transfer between the two proteins, as well as what the transfer complex looks like. And I think you might wonder why this is important, but again, fundamental chemistry here is of great interest to a lot of different fields of research. So just to reiterate, for those of you who weren't here yesterday, you never find copper or any transition metal really free in the cell. There's usually something that is holding on to it, even if it's a non-proteinaceous ligand, it's never left alone. And these metalloprotein interactions are everywhere. And we really don't understand a lot about copper, actually. It's relatively new, evolutionary speaking. Yes, it's new to the crowd. And there weren't a lot of ways to really look at it spectroscopically, particularly copper-1, until we had more technology to be able to do that. So I like to think, how does a metal ion move directionally, like from one place to another? You know, this is kind of my thing, right? How does it move from chaperone to target? I like to think about it as a game of thermodynamic hot potato. This is a game that people used to play when we didn't have Twitter. In the 1800s, you would heat up a potato and stand in a circle, and you would see how long you could hold on to it, you know, how macho you were, and then you would pass it along, right? And so that's basically what I like to think about when it comes to metalloproteins and how they transfer things. And yes, I found a hot potato. But here's the scientific representation. Beautiful, beautiful work by Lucia Banchi in Italy. And so what this is showing is essentially the delta G values. It's a three-dimensional, it's very fancy, right? So you have your delta G's here, and then you have the delivery pathway. And then over here, you have some of the more, I would say, vital proteins and systems that are needed to move metals from one place to another, okay? So I just want to point out, this is glutathione. It has essentially the highest affinity for copper. And so it's just GSH, so it has a sulfur there, it has the highest affinity, and it's used a lot in the cell, but it's often when you have too much of something, it really starts to upregulate that so you can grab it as quickly as possible. Then here we have, I would say, I think this is more human or mammalian. Yeah, this is for the trans Golgi. And then we have HAH1, COX-17. These are all just various metalloproteins that grab on, and they're about relative affinities in terms of free energy. And down here we have metallothionein. So this is that hot potato. Basically, it's going to move directionally based on the thermodynamic parameters. But that all makes sense. It's going to move from one place to another. Okay, but what about when the thermodynamic gradient between the target and the chaperone is very, very shallow? And this is the example that came from the work that we were doing here. So just to quickly recap, this is the periplasmic vacuum cleaner. This is what I spent some time trying to understand. It's an E. coli. And when you have copper or silver stress, what happens in the bacteria is that this is made in excess. These little proteins here, they come and grab the copper or silver very quickly. And then what the work I had done to show, I'll wait for that. The work I had done shows that perhaps CUS-F delivers its cargo to CUS-B. That affects some kind of conformational change that opens up the CUS-A pump and allows for silver and copper to be transported through that pump all the way out. And then it most likely is oxidized on the way out. And the thing was, these two proteins seem to interact very intimately, and they seem to communicate a copper state to the cell. And this is the one slide that describes everything I talked about yesterday, which is that I use selenomethionine labeling, and here it is here, to provide a powerful internal X-ray absorption probe to be able to look in turn at what is going on and see what the role of this CUS-B is. Since these two have to interact in some way to activate the pump, there was a lot of argument as to whether the CUS-B was a relay of some kind, like maybe it went from here to the pump, or perhaps it was the other way, which is that this is needed to bind metal first, that activates the pump, and then CUS-F is able to deliver its cargo over here. So this was a big argument. And again, I'm sorry if you missed yesterday, but in 2014, what I did was essentially use selenium labeling, since each of these proteins had methionines that we could label with selenium for a better scattering atom in X-ray absorption. I loaded one of the proteins here, CUS-F, with copper. I loaded the CUS-B with silver, and then included this basically to start the reaction between the three of them. And I was able to essentially recreate the pump activity and show that CUS-B was a switch. It had to bind metal first, and then CUS-F was able to deliver its cargo over to CUS-A, and this added some element of control. So just quickly, we hypothesized that when the copper concentration is high, CUS-F loaded copper to CUS-B, that activated the pump, and then CUS-F was able to go in and do this. But if copper concentration is low, which I showed in the experiments from yesterday, if CUS-F is empty, say we've exhausted all the copper that's in there due to the copper stress in the periplasmic space, then CUS-F will begin to nip back those bits of metal ions and slow down the pump incrementally. And that's important. You don't want it just on all the time. So this is where we get to what's going on today. So turns out these two have nearly identical copper 1, silver 1 binding affinities. If you do the isothermal calorimetry, you see that they have exactly the same affinity for their metal. So why then is it moving directionally from one to the other if that's the case? There has to be something more going on. There has to be, otherwise they would just sit there. There's just something going on there. So if one partner is empty, the full one would transfer exactly half of its metal and then it stops. If both are full, they do not exchange metal. And so how can we understand transfer between these copper 1 protein partners or silver if they have a shallow thermodynamic gradient? They have very similar binding sites. So we have a MET-HIS and a tryptophan cap, which I'll talk about. And then the CUS-B has a MET-MET-MET. So really it's not too different, but just different enough. And yet if you measure their thermodynamic parameters, they're exactly the same. So we have these similar binding sites and they're silent. We're dealing with copper 1 or silver 1. That's mostly silent to any in-lab methods that you might have. So what can we do with these in vitro experiments to tell us about CUS-F and CUS-B metal exchange? This is what I wanted to try to do. Some of this was trying to do it just because it could be done. And I think that's what spectroscopists and scientists like to do. I had been trying to use rapid freeze quench on a project for quite some time, coupled with XFs. And it's really difficult to do because a lot of people do, I'll explain RFQ in a minute. But anyway, I would like to see if I could mix these two proteins together, one with the metal, one without, maybe use the selenium labeling, and try to see if I can find an intermediate complex. Let's see what happens when they come together. Is there some kind of sampling going on, which is helping to move it along to one side or the other, one protein or the other? And it would be even cooler if we could get a kinetic profile of some kind, get the time course of this. And then the other thing I'm going to talk about is exploiting the lone tryptophan residue in the CUSF active site to do some stopped flow tryptophan fluorescence copper transfer experiments. So we're going to combine XAS with some stuff in the lab. So rapid freeze quench. So this is me teaching. Can we turn off these lights actually? Thank you. So rapid freeze quench is kind of like chemical quench. And if you haven't seen chemical quench, maybe think about stopped flow. You have a number of syringes, one with something that will react with the other. And then depending on your detection, you can use all kinds of things, a photodiode array, you can use a photomultiplier. There's all kinds of different ways you can track things. However, for XAS, you want to prepare something in the lab and then you have to take it to the synchrotron and you don't know if it worked or not, okay? So this is Stuart. This is my Kintech rapid freeze quench. I like to give my things names because students will treat them better, I find. They're like, don't hurt Stuart. It's different. I don't know. I don't know why. They're like, oh, but take care of Stuart. So anyway, what I did is that I put CUSF in one, CUSB in the other. I did a bunch of different conditions, so I'm just going to keep it simple for now. One of them had the metal and one of them was selenium labeled, okay? And I did that in the mirror image. So not only did I do CUSF, for example, with copper and the selenium label, but then I did the opposite experiment where CUSB had the selenium label and had the copper and every permutation therein. And so you mix these two together, it goes into an aging loop. That's actually the displacement is what allows you to have the time change in this case. It's a little bit different than other processes. And then it goes into liquid ethane because liquid nitrogen actually is boiling your sample. So you can't use that. And it also serves as a pretty good glassing agent, actually. Strangely enough, you get around a lot of the problems that you have with glycerol, something about the way that it sprays out. And so then you quench the reaction, and that's our basically our liquid ethane doer here with a specially machined little thing, and then it sprays out and you use this little tamper and you put it into a tiny little XAS cell and you hope for the best. And so my goal, this really hasn't been done a lot. People have done it where you mix the same thing together and you do it over and over. Like you do copper loading into a protein and you really just look at one time point and you're trying to isolate something. But this is actually trying to look at the entire time course. And then you could also get kinetic and structural data, which would be so cool. So again, just quickly, what I want to point out is this is what it looks like in the XAS if you, or the XFs, excuse me, and we love Fourier transforms because they're nice to look at. But here what we have is what happens when you just at real time in your laboratory mix the selenium methionine labeled CUSF with copper. So it's ready to go, the little green protein. It's loaded up and it's labeled. And then you mix that together with the APO or empty CUSB. What you can see is that the selenium copper goes down by half. Remember, they transfer exactly half. And you can do the opposite and see that exactly half will go from one protein to the other if you do the other side. And just to show you, this is the aposelenomethyonine CUSF. And if you're curious, this actually seems to be a bunch of carbons involving with the selenium, what it sees. So it's something kind of weird there. So I generally will fit that in. Now this is going to look like shit, but it's not. Just want to say this I have done in, I did it 4 times, like 4 different beam times. Got the same results every time. And so, you know, if you came into your PI with a kinetic trace like this, they'd just laugh and they're like, what are you doing? You're not catching the fastest part. And so I did everything. Went to 4 degrees. I tried to change the viscosity. You do all these different tricks to try to slow things down. And no matter what happened, what we had is this extremely fast first phase within diffusion limits, actually, over and over. And then we had a slower phase, okay? Over and over this happened, and I did it with both the CUSB going to CUSF and vice versa. You get kind of the point. And this was done by undergraduates and Stuart and me.
Okay, so I kept doing this and I thought, well, this is actually really cool because yes, we're getting a time course that at least tells us it's biphasic, just simple chemical kinetics. We're not doing any kind of enzymology here. But also this is the selenium edge and it's giving you selenium copper occupancy, okay? From the point of view of whichever protein is selenium labeled. That makes some sense? It's a little weird. We can always talk about it after. I'm going kind of fast. But you can also switch and look at this a different way by analyzing the x-axis. And you can switch to the copper edge and see what's going on from that point of view. So we actually get a lot of information, but this is the one that you choose to put in your paper because it's at least giving you some idea. You might be curious about the effect of the selenium label on the affinity of one protein for the copper. And in fact, we do see that there's a slightly higher affinity for the copper to be in cusp-B. Okay, it looks a little bit crappy, but this happened over and over. Essentially, when you have the label on the cusp-F, it's not able to grab the copper quite as strongly. This makes sense because it's 2 methionines and 1 histidine in that active site, whereas the cusp-B has 3 methionines. And when they're labeled, that does create more of an affinity for copper. But this is just going to come back later. This is not really to prove any point, except to say, when you look at the copper selenium shell occupancy, that was 15 minutes. It's done. We see that more copper moves to cusp-B than to cusp-F when cusp-B is selenomethionine labeled. Okay, just keep that tucked away. So if we switch, we saw what happened when we looked at what selenium saw, which is okay. We see half of the transfer and we get it in real time. That's cool. But it really does matter on where you are and what you're labeled with. So take home from all of this is when you have the label on the cusp-F, the little green guy, you end up getting this particular ligand set. This is cusp-B here. This is a QMMM prediction. So you have the 1 methionine coming from cusp-B, and this is what we actually were able to simulate in the x-axis as well. And then we have 2 ligands coming from the cusp-F. But when you have the label on the cusp-B, you see that more ligands are coming from the cusp-B, 2 methionines, and then you're only getting 1 methionine from the cusp-F. So we have 2 populations. So this is great to know. Cool, cool. At least we know we have some structural information now, and we have somewhat of a kinetic trace. But there are, not enough time points that I could catch. I tried over and over. It's just so fast, no matter what we tried. So we needed to find a new vantage point. So it turns out cusp-F has a very unique tryptophan. It actually acts as a little cap, and it participates in a cation pi copper-1 tryptophan interaction, and it basically seems to protect the copper-1 from solvent. If you think about it, that cusp-F is running around doing a bunch of stuff in the cell. And so we're looking at the tryptophan fluorescence report on copper-1 transfer in a different way, and this can be done in the lab. So I don't know, do I need to go into fluorescence? I don't necessarily, thank God, okay. So here's what happens. It's actually really cool. So this is cusp-F just in the fluorescent spec in the lab. So we don't have to go anywhere for this. This is how bright it is when you excite at 295. That tryptophan is really bright in the empty protein. You can add copper-1 in aliquots and you see that it quenches it quite beautifully. Now this was done previously by O'Halloran and co-workers, who does a lot of zinc work actually. But when I did this, I didn't know any better when I was doing fluorescence, and I went out to about 600, and you do see the first, what do you call it? Reflection harmonic. If you go out this way, you'll see the first harmonic of what you're doing over here, but we ended up seeing this little thing over here, and it was very strange, and it's 1-to-1 with adding copper, both this and that. So we know it's stoichiometric with adding copper. We can use this to look at copper transfer. And just as a cool nerdy thing, this is a cation pi charge transfer. We did the lifetime measurements, and it's just really neat. You never usually see something in fluorescence way over there. And I guess you would call it luminescence. So can we use this to track copper-1 transfer? Let's see. This is where this is handy, because if this is what naturally occurs in the cusp-F, then you should be able to see that when the cusp-B delivers its cargo, that you get that same feature, so that it's not some kind of I don't know, maybe solvent thing that's happening. And so we saw the same kind of effect when you added cus-B to cus-F, you see the same little peak. So we can use this to look at transfer between the two proteins, okay? So can we monitor it in real time? And what I want to point out is that you have to lift up that little tryptophan in order for the two to meet and be able to transfer their metal. So when you're watching the stop flow, I want you to keep in mind, this is not the same process as the XAS. This is actually watching that tryptophan flip up and then take its cargo and move away, okay? So I don't really need to talk about this. I just sort of moved some things around to make fluorescence a little bit better. It's really hard to do it with stop flow because you're mixing so much, you get a lot of quenching. And so what I ended up doing is rearranging, getting an LED source with really a lot of photons, and then selecting, using my monochromator, one wavelength. So just to remind you, we're looking at that tryptophan, not necessarily the sulfurs or anything like that. And by golly, we caught the fast phase. Very exciting. And it's really freaking fast. So this is essentially watching the tryptophan quench quickly as it takes on some of the copper, and then it sort of stalls. It goes into this slower thing. And this does include a small photo bleaching slope. With that many photons, you're kind of breaking your protein apart. But this is actually the slower phase, taking that into account. So take home, we still have this biphasic situation where they encounter each other. You get this quick transfer of some of the protein or the copper, but then they sit there for a while before they actually transfer it. And this is me showing that you can do this with the selenomethionine labeled one, that it can be done, that you do change the rate a little bit, but it's not that bad, and it's consistent and in scale. You can do it with other kinds of proteins. For zinc, for example, most metalloproteins have a tyrosine or a tryptophan, something kind of spectating. I'm not sure why they're there, but if you have a protein that has some kind of a aromatic residue, talk to me, especially if you're working with a silent metal, because this really does work. And so I just want to say that Kenny's in silico models, actually, which he had come up with before we did this work, and I didn't even know about it, agree with this sort of idea that you have this flipping up of the cap. The two proteins come together, they transfer quickly, but then they hang out for a while before the cus-F kind of moves away. Seems like a communication of some kind to me. And then the other really cool thing was that he predicted two different populations for that capture complex.
Sorry to interrupt. Complex. Has anybody followed this by NMR? Like I just recently
learned. It's a great question.
And I recently learned there's this really cool way of labeling tryptophans with fluorine.
Yes,
and
yes.
doing fluorine
we talked about that. We
So
talked about that.
Have you have any?
I have not done it.
Because I think you could see this
A little
very
bit more.
nicely, right?
Yeah, the role of the tryptophan for sure. So, no, when they came together. So what we had done is, I didn't mention this, when you saw that rapid freeze quench, you have your one spot there and then you had kind of these going over here. This indicates kind of a steady, like this is where they're just hanging out. And that's what we use to simulate what was going on with these two populations. And depending on where the label was, we would have either more coming from the cus-F or more coming from the cus-B. So the major predicted populations that Kenny had come up with his QMMM was that you had we had a major predicted population, which is exactly what we saw, that two of the METs were coming from cus-B, one from cus-F. And then this was the next one down, is that you have a population that has one MET from cus-B and then the other ligands from cus-F. And all this is to say, I'm gonna stop here. All this is to say that it's supported, we're getting this idea that for these shallow thermodynamic gradients, that there's a lot of ligand sampling going on. And all of a sudden, one of those methionines must kind of hit the sweet spot. It's kind of reaching in to the other guy. And that it's just sort of finds the right spot and bam, it takes it away. And then it can finish what it's doing. And this is just something to keep in mind when you're looking at these kinds of protein systems. It's been shown before by other groups. Amy Rosenzweig did some stuff with, I think, ATOX1 and some other systems like that. But we're getting able to look at these little secret handshakes. And I think it's really cool. So that's the first part. Any questions on that? It went really fast. Yeah, I did. You saw me yesterday, like relax. Today, I'm like, okay, business, let's go. Because if not, I can go on to what people really want to hear about, which is, okay, great. So, maybe you have a protein or a molecule that might bind metal. You know, you could, like, I just want to say, this is for everybody. You know, beam time is for everybody. You just have to figure out how to do it. Maybe you know a lot about your metalloprotein, but it won't crystallize. Okay. Maybe you know a lot about the metalloprotein, but it will not crystallize. Or possibly you simply have an interesting biological system that in some way needs XAS. No matter what, you're going to need to write a proposal for that or work with somebody who can, but you are not alone. We are here to help you. This is why we had this workshop, is to excite you with the possibilities and say that it is within your reach. Okay. This is a lot of words. Don't read them. I'm just going to talk to you. Okay. So to prepare for a competitive proposal, this is basically how it often goes. And this is really informal. Okay. I'm just kind of telling you the reality here. It's important at first, say you have no experience, your PI doesn't really know a lot about this stuff, and you really want to get some interesting XAS. You have to pique the curiosity of someone who does XAS or, you know, any kind of X-ray science. You have to go to Thomas and say, oh my gosh, I have this thing and it's really, really cool. And I think your technique could work on it. And you need to meet with them in some way and give them a very short sort of pep talk on what you're doing and why you need it. Okay, because everybody says they want to use XAS or X-ray science for something, but you want to show why it's exciting. Okay. And it isn't that hard or too much time taken from someone who does it to try something out for you. Okay. So this is if you want to try to, like, you could try to do it all alone, but this is a nice way to do it. And you know people now. That's why the workshop was here. So send a sample. They're like, cool, this looks really cool. Okay, I'll send a sample or two with you. You got to be careful because you wrote a proposal for a specific thing. Thomas or I or anybody, you wrote one for a specific thing, but it's okay for you to run 1 or 2 samples just to see if it's interesting. And that hopefully will work out. Send some replicates, follow their instructions for sample prep exactly, and send a detailed sample list with what metal concentrations, pH, buffers, every bit of information that you can think of, and anything that went wrong while you were trying to make it. It's fine, we can still run it, but just tell us, oh, it didn't quite go as well. When you get the data, please do not argue with the data or ask the expert to fit something that is not there, unless you have some other technique that without any doubt indicates that, you know, it supports your claim that there should be something else there. Okay. XAS or X-ray science cannot just make things up for you. And I find that a lot of times people will say, oh, I think you can finally show that that ligand is there. It's just not that way. It could just, you know, whatever. But even if this doesn't work perfectly and you have some crappy data, it doesn't matter. You went through the process and now you have some preliminary data that you can put into a proposal. I want to point out that when you start out to write these proposals, it's okay to borrow completely from an existing proposal, okay? This is not meant to be a standalone grant or something like that. It really, I mean, it's worth about $65,000 in beam time. It's worth a lot. It is a grant and you should be proud if you get a beam time proposal accepted. However, it's totally okay to just take the figures or the stuff you have and make that into your proposal. It's going to look really good. And so keep it simple, which sounds great. This is easier. Like, it's not as bad as you think. 2 to 3 pages, understandable to a general scientific audience. Figures are very helpful. Realistic and narrow scope for now. And then why is XAS crucial for your project? The people reviewing are not going to really know your particular subfield or that zinc is really cool or anything like that. But what they are going to know, why is it so important? Okay, it doesn't have to change the world, but it has to be like, this data will really help us to get to that next level. And do not include anyone in the proposal that is not giving you permission to do so. So don't just put, I keep pointing out Thomas, but don't just put Dr. Kroll's name on there and be like, oh yeah, well, I know him and he'll see it and it'll be fine. So don't do that. I don't know why that happened. Okay. So you submit the proposal. And I'm not going to show you the SSRL website. I'm partial to SSRL. It's where I go. There are many other beamlines. The proposal, once you send it in at the appropriate times, there's about 4 times a year. So don't feel like you've missed out if you didn't get it during the one cycle. You can send it in many different times of the year. Your proposal will go to the people that you suggest. You can give some names. We have names. You can put our names in. Yes.
I always wonder.
Yeah, I can tell you anything. Yeah.
When I suggest 3 reviews,
Yes.
it will show them. I thought
It will
might
be,
not, but it
you will be, we will ask those 3 people, unless we have reason to believe there's a conflict. They may decline. And then from there, but generally we will ask those people. You will get a review from those people, especially if they're in the field and they know, because one hand washes the other. We all have to do this. This is the game. We all review things and we all get reviewed. And so it's the same with the proposals. It is good to know. And you can tell them who you don't want to put in. That's important too. There's times when you're like, actually, this is kind of a big contentious thing and I just really don't want to deal with them saying that my stuff is BS. That's okay. It will go to some people that the panel suggests. And at time, those of us who are on the SSRL review panel will review it because we have expertise in it. So generally, if something has copper or some kind of bioXAS, I may volunteer to look at that. So at SSRL, at least, you will get a review score back, no matter what, and comments. The difference between this and many other kinds of proposals where you just have to get mad and drink a beer and say, these guys don't know what they're talking about, is that you can and should respond to those comments because it actually is just to help us understand what's going on. So especially if you're new to this, it's okay. You can say, gosh, I didn't know that 900 hours of beam time was too much. You know, it happens, right? Because you're supposed to like multiply the shifts and figure it all out. And it's supposed to be over 2 years. And you don't know at first how long you're going to need. And so this is just coming from the SSRL website. We have this proposal review panel that I am on that is on a scale from 1, should like get this on the beam right now, to 5. Yeah. But generally, the scores tend to run from 1 to 2.5. It never gets to 5. We're probably going to revise the scoring thing. So if you get something in the 1 range, I can tell you that's probably great. And even if you don't get first dibs, you will get some time. Okay, we want the National Lab, the DOE wants you to use this. We want more people to be using biological or any kind of access to these beamlines. We're not trying to keep you out, but it is hard to get over that little activation hump. But now you know people, okay? I have a bunch of very comprehensive resources. I am happy to email these to you. I know you're squinting at it, but I just put them there so that I had them all together. There are some great podcasts and videos literally on how to get started. Kathy Knotts does a great job of talking about all these things. My favorite is, there was one from Stanford that's like, how to wow. It really is great. Once we're no longer recording, I will show you some proposals just out of respect to the people that didn't give me permission in the time that I had. But again, those of you who are watching from other places, just email me and I'm happy to share those with you or discuss them with you. Okay, it's going to go on YouTube, so I don't want to do that. And I have a lot of other things I could talk about. I wanted to leave it open for questions. So with that, I will stop. But I hope what I impressed upon you is that it's not impossible. Once you get that preliminary data, then it starts to snowball, especially if you get something back. And then you can say, we tried a little something, it's promising. As you've said before, you mess it up about 3 times before you get it right. That's expected and part of the process. And, so it's okay they know it's exploratory. And now you know people, you can talk to me. If you have a sample or you want to try to do this, just call me. I'm close. My flight to SSRL is an hour. Okay. So think about that. Okay. I'm going to stop there. Questions?
Yeah, I completely agree with everything.
Great.
Like the previous presentation was essentially that. Like one time I took the trip to build Fegan tubules with me, and it's like, oh shit, we can measure that. That's
Yeah.
interesting. Oh, and it looks very different each time. Wow,
Yeah.
it's reproducible, and that's how the entire story goes.
And the panel's really open to it. We talk about it. We're like, yeah, this is cool. We should give them a try. We may only give you half of the time you wanted, but it's okay. And now you can actually look at it remotely, which is a big deal on some of the beam lines.
So, questions?
How do you know how much time to ask for?
I swear I had written that down and I don't know. That's really weird. I could have sworn. Anyway, generally, so it's 8-hour shifts, 3 shifts a day, which means you better be ready to work at night. And usually we give that to the most junior person so that you can panic when you accidentally like leave the helium on, or like it's a good learning experience. But we will always I have to get up in the middle of the night and be like, oh, you just didn't turn off the vacuum. Okay. Anyway, so you have to think about that. I would say for a preliminary experiment, what you can also do is ask me how long it really, like, say you send like a sample or 2 with me, and I see that it's pretty dilute, or that there's something that's going to have to happen, or wow, you really messed, this doesn't look good at all. But if it takes, hopefully it doesn't take more than 3 hours a sample, depending on what you've got. So then you have to think about what you need. The less time you ask for as a newbie, the better. There's also rapid access proposals, which aren't just for emergencies. They're for exactly this. I have this thing, really curious. Can I just have 2 days and just show you that I can get something? And so I asked for way too much time. I can show you that later. And they were like, we're not gonna give you this time. But then I just wrote back in the comment, I was like, hey, newbie here. What time do you think would be appropriate? And then they gave me an adjusted time. We're real people. That's the thing. Yeah. So I guess it's not a hard and fast rule, but assume 3 days of beam time per cycle, and then multiply that by 2 years. And the cycle is what, there's like 9 months of beam time.
Yeah.
So 9 months, 2 years, you might get time maybe twice a year. If you get like a moderate or mediocre score, that still will give you time.
Thank you.
Progress, not perfection.
And it all depends on the concentration.
Yes, absolutely.
It's high, yeah, like
Okay, should we stop recording and then go ahead and look at some proposals? Again, contact me if you want more information on proposals.
I can also add like
Please,
a
yes.
comment to
Yes.
the proposals.
It's not just for me,
And
yeah.
adding on to what Kelly said, basically when you write a proposal, there are 2 types of proposals. There's a grant proposal and a green proposal. Right? Like in the the grant proposal, you want to build your big picture and explain what great science you want to do, and that's more important. In a beam time proposal, you also want to say why it's super